All right. Hi, everyone. I'm Sarah, and let me just share screen. All right. So I'll be taking your neurophysiology lecture for today. Before we get started, if anyone has any questions during the lecture, just feel free to pop it in the chat or unmute yourselves. Also, my student email is down there, so if anyone has any questions after the lecture, feel free to email me as well. So this is kind of the synopsis of the lecture, and it looks like a lot, but I've kind of broken it down a lot as well. So we'll be starting with your neuron basics, and then we'll go into your resting membrane potential and your action potentials. We'll look at a bit of the refractory period, your graded potentials, and some other things after that related to action potentials and graded potentials as well. And then we'll go into synaptic transmissions, and we might take a short break after looking at receptor types, and I'll try and have a look at the chat then for questions. And then we'll look at the organization of the spinal cord, like your gray matter and your white matter. Then we'll look at your reflexes, the different types of reflexes, then the different types of nerve fibers before we go into our favorite ascending and descending pathways. Our last topic is about the autonomic nervous system, and we'll finish the lecture with a short quiz. So if you guys could participate in the chat or direct message me in the chat as well. All right, so I'm sure you guys might have seen this diagram a few times. So if you have a look at the top, we have your dendrites, which is where a nerve receives a signal, and you start to induce a response along the axon. At the very start of the axon, you have the axon hillock, and that's where your action potentials start. And then the axon is where the signal is transmitted down, your electrical signal. And these kind of gray box-like structures along the axon are your myelin sheath, which insulates the axon and helps to speed up the rate of conduction. In between the myelin sheath, you have what's called your nosoverandia, and there are spaces where you find ion channels that allow for ion exchange with the extracellular environment and during the propagation of a signal like an action potential. So these are the different types of neurons you can find. We have unipolar neurons, multipolar neurons, and bipolar neurons, as well as pseudo-unipolar neurons. The two that you need to remember in particular are your multipolar neurons and your pseudo-unipolar neurons. So your multipolar neurons are the neurons you can find in the central nervous system, whereas your pseudo-unipolar neurons are the ones that you can find in your dorsal root ganglion, where your sensory neuron bodies are found. And we'll come to that later as well. Other cells of the nervous system, are your neuroglia or glial cells, and these are non-neuronal cells, meaning that they don't conduct electrical impulses, but in general terms, they help to maintain homeostasis, and they're the cells that also form the myelin sheath that we were talking about before. They help remove debris, waste, and pathogens, and they also provide support and protection for the neurons. So there's a lot of cells here, but I don't believe you need to remember them all. I guess the two that you might want to remember are your oligodendrocytes, which help myelinate the axons in the central nervous system, and your Schwann cells, which do the same job in your peripheral nervous system. So our resting membrane potential is the steady potential of an unstimulated cell, and it's created by the difference in voltage between that extracellular environment and your intracellular environment. And this difference is around negative 70 millivolts, with the intracellular environment being negative relative to the extracellular environment. RMP is affected by three factors, which we'll go through one by one. The first being the diffusion of ions down its concentration gradient. The second being the selective permeability of the plasma membrane through specific ion channels. And the third is the electrical attraction between cations and anions. So the first factor being the diffusion of ions down its concentration gradient. So if we draw a plasma membrane, and then we look at your potassium concentration, there is a much greater potassium concentration in the intracellular fluid compared to the extracellular fluid. And so you have potassium flowing outwards, and then contradictory to that, we have your sodium concentration, which is greater in the extracellular environment. And so you have flow of diffusion of the sodium into the cell. Something to note is that potassium is at equilibrium when the voltage of the system is at negative 90 millivolts, whereas the equilibrium for sodium occurs when this voltage is at positive 60 millivolts. Now, neither of these values are the resting membrane potential, which is at negative 70 millivolts. And that's because we have other factors contributing to, that's because we have to combine the two concentration gradients, and then there's other factors as well. And so that's why RMP, if we consider other factors, which we'll get to next, that's why RMP is closer to the concentration gradient for potassium than it is for sodium. And that's because of these leaky channels. So leaky channels are the ones that, this one specifically important for regulating your resting membrane potential, and they have an intrinsic rate of opening and closing. Some people might say that in some other resources, you might also see that leaky channels are always open. And so your potassium leaky channels are far greater in number, around 25 to 30 times more potassium leaky channels than your sodium leaky channels. And so your membrane is more permeable to potassium. Other ion channels that are important in not necessarily your resting membrane potential, but say in generating your action potentials that we'll get to in a bit, is your voltage gated channels, and these open because of changes in the transmembrane voltage, so either depolarization or repolarization. Your ligand-gated channels are channels that require ligands, like neurotransmitters and hormones, to open. And your final ion channels are your mechanically gated ones, which change because of a physical change in the environment, things like changes in pressure, touch, stretch, and temperature. So if we look at the final factor affecting your resting membrane potential is the electrical attraction between cations and anions. So even though we have a lot of positive charge from the potassium, we also have a lot of negatively charged large proteins that cannot cross the plasma membrane, and that's what makes your intracellular environment more negative than the extracellular environment. So because of these negatively charged proteins and other anions in the intracellular environment, you have a pool of some of the potassium, the positively charged potassium, back into the intracellular environment. And so this is the potassium electrical gradient. On the other hand, you also have chlorine, negatively charged chlorine in the extracellular environment, which pulls some of the sodium back out. And so this is the electrical gradient for your sodium. One final thing is your sodium-potassium ATPase pump, and this acts to transport your sodium and potassium against its concentration gradient. And so it requires ATP. It's actively transported. So what occurs is you have 3 sodium going out into the extracellular environment and 2 potassium going into the ICF. And this action counterbalances the rate of passive transport via those leaky channels. And it's important in maintaining the high intracellular concentration of potassium whilst maintaining the high extracellular environment of sodium. You'll see later as well that it's also important in returning the membrane potential back to resting membrane potential following your action potentials. Before we get into action potentials, does anyone have any questions up to here? Let me have a look at the chat. No? Okay. So the terminology we want to know for action potentials is things like depolarization, which is when the resting membrane potential when the membrane potential increases above RMP, so it becomes less negative or more positive than RMP. Repolarization occurs when your membrane potential returns back to that resting state, whereas hyperpolarization occurs when the membrane decreases below RMP.
Can I just ask a question before we move on, please?
Yep.
So with regards to the sodium potassium ATPase pump, is that an entirely separate thing than the electrical gradients created for potassium and sodium, or are they interlinked?
So they're interlinked in the sense that the electrical gradient is I think you asked about the electrical gradient, but it's linked to the concentration gradient rather than the electrical gradient, because it's not really pulling in. It's not really, because electrical gradient is kind of the pull between cations and anions, whereas this one, it's not. You're exchanging your positive ion, so there's not really any anions involved in the transport of sodium and potassium, if that answers your question.
Yeah, okay. So yeah, okay, thank you.
All right. Okay, so our next, so how do we generate your action potential in the first place? It's due to an electrical stimulus at the very, it's due to the electrical stimulus at the start. And the response to this electrical stimulus is an influx of sodium through voltage-gated sodium channels, and this makes the intracellular space more positive. So it depolarizes that intracellular space. One thing to note is that action potentials can only be generated when a certain threshold is reached. So the increase in membrane potential has to be sufficiently significant. And I believe the value that you're taught was -55 millivolts for the threshold. And so it's described as an all or nothing response. So there's 3 states that the voltage-gated sodium channels can be in. They can be closed during RMP, but at this stage, they can also be opened to stimulate your action potentials. They can be opened and activated, and this allows your sodium to enter the cell. But then after this, during the period of repolarization, they become inactivated. And this is, in this stage, your sodium cannot be opened and you can't let any sodium in. And this is what would cause what's called a refractory period. Before we get into your refractory periods, you also have one other voltage-gated channel that's important, which is your potassium channels. And this allows potassium to flow out, causing repolarization. So you have a buildup of positive charges when the sodium flows in, but during repolarization, these voltage-gated potassium channels open, and your positive and your potassium positive ions flow out and cause, decrease of your membrane potential. The rate that these potassium channels close is very slow, and so it causes a period of hyperpolarization following the repolarization. So it goes even below the resting membrane potential. So if we were to look at this step by step, at the very start, you have your resting membrane potential, where there's more potassium permeability. And then this is followed by a period, and actually before the period of depolarization, you have you can see here that there's some failed initiations, and this is when the depolarization stimulus does not reach the threshold. After that, you have your period of depolarization, and that's when the action potential is generated, and there's more sodium permeability. During repolarization, however, you go back to having more potassium permeability, because now the voltage-gated potassium channels are open, and the sodium voltage-gated channels, they become inactivated. During the hyperpolarization state, you have more potassium permeability still, and your potassium voltage channels are slow to close, whereas your sodium channels start being resetted to a closed state, so that they may be opened again for another action potential. This kind of leads us into our refractory period. We have two types of refractory periods. So during your absolute refractory period, no new action potential can be started, no matter how strong the stimulus is. And that's because your sodium-gated channels are either already open or they're inactivated. And this prevents your action potential from spreading bidirectionally. Your relative refractory period is when your inactive voltage-gated channels reset to that closed state. But because of the slow rate of closing of those potassium voltage channels, a larger stimulus is required to combat this, to start an action potential, compared to when you're starting at resting membrane potential. Your sodium-potassium pump also plays a role here. It exchanges your sodium from the intracellular with the potassium from the extracellular again, and this restores your resting membrane potential back to -70 millivolts. I think I have a few questions in the chat. So what type of membrane channel is responsible for the initial change in the membrane potential that causes the sodium voltage-gated channels to open and trigger an AP? I believe it's still your voltage-gated sodium channels. So what happens is we'll get to graded potentials in a bit, but graded potentials, when a certain number is reached, a certain voltage is reached because of those graded potentials, it can cause like the opening of a lot of more sodium channels, and that causes the influx of sodium. You can also get like inhibitory or signals from, say, like your neurotransmitters, and that's what causes those signals are what causes perhaps the opening of those sodium channels. Another question: wouldn't restoration by these sodium potassium make it more negative, since 3 cations go out and potassiums go in, making it more negative? I'll get back to you on that one. So hyperpolarization. Okay. So you have sodium, because you have the sodium influx going in, and so you want to pump back some of that sodium back out, and your potassium that's gone out during that, through the potassium voltage channels, you want that to be pumped back in. I hope that answers your question. All right, so if we look at the resting membrane potential in this diagram here, during the very start, when you're at resting membrane potential, a smaller stimulus is required to start that action potential. But during this absolute refractory period, right before the period of repolarization, your sodium channels become inactivated, and so no matter how strong the stimulus is, you can't start another action potential here. During the period of hyperpolarization, your during the period of hyperpolarization, there's you can start another action potential here because your sodium channels have returned back to its closed state, so they can be opened again. But again, because it's at a lower stage than at resting membrane potential, a greater stimulus is required here. But as it goes back closer to resting membrane potential, you can return back to, having the normal stimulus for your action potential. So this diagram kind of tells you how the action potential is propagated down the axon. So, like I said at the start, the action potential starts kind of at the axon hillock with the influx of sodium, and this causes the opening of sodium-gated channels on the next adjacent membrane, and this continues down the axon. In this diagram, you can see that the charge doesn't spread bidirectionally because as it depolarizes the next membrane, the previous membrane, which is undergoing repolarization, has its sodium channels inactivated, and so you can't have sodium channels opening and sodium influx in the previous membrane. And so the action potential only goes in, and that's what is the absolute refractory period. So basically, action potentials can't occur on the membrane that's undergoing repolarization. Any questions up till here? Does the magnitude of the action potential decrease during the relative refractory period, or is it just the largest stimulus that is needed to produce an action potential? I believe that if we go back to a few slides, the magnitude does decrease during the relative refractory period, but as you get closer to resting membrane potential, it should go back to normal. And that's because I think the response to a stimulus is all that exceeds the threshold required for an action potential is always going to be the same. And so because you're starting at a lower point here, it's not going to be as great. If there were no potassium voltage gated channels, why would the membrane potential return to normal? More slowly rather than not return at all. So I'm not sure what you mean by if there were no potassium voltage gated channels.
Oh yeah, I'll ask now.
Yeah.
Sorry. So if there, let's say there were, and we in the normal neuron we have Na+ and K+ channels, right? If the K+ channels weren't there, why would the action potential why would it spike infinitely? Why would it not spike infinitely and not return to normal, rather than spike but then depolarize gradually?
I think because you still have your leaky channels where because the sodium becomes inactivated, so you don't have a continuous inflow of the sodium. So the action potential doesn't keep going upwards. It doesn't keep depolarizing. And also, if there were no potassium channels, you still have your other channels say your leaky channels are causing the efflux of potassium outwards as well.
Okay, yeah, that answers it. Thank you.
All right, no worries. Okay, so our graded potentials are local smaller changes in the membrane potential, and they can only spread over short distances. The spread is bidirectional though, compared to unlike those action potentials, and a lot of graded potentials can stimulate an action potential. So they die out over short distances again, and the spread is decremental. So this where the change in voltage at the point of that stimulus is greater than the change in voltage further away from the stimulus. So it gets smaller and smaller until it dies out. Your EPSPs and IPSPs are small graded potentials that either causes depolarizing through an influx of Na+. So EPSP is your excitatory postsynaptic potential, yeah. Excitatory postsynaptic potential, and it causes that influx of sodium, and it brings it closer to the threshold and makes the likelihood of an action potential firing more likely. Your IPSPs is basically the opposite of that, and it occurs when an inhibitory signal causes hyperpolarization of the neuron, and it decreases the chances of an action potential firing. When you have those like added EPSPs and IPSPs, you have what's called summation. So what happens is multiple neurons can terminate onto the dendrites of a postsynaptic neuron, which can cause either that excitatory or inhibitory response. The summative effect needs to exceed a threshold to trigger an action potential again. And if the EPSP and IPSP are of like similar or like the same magnitude, they can cancel each other out. So there's two types of summation. You can have spatial, where a lot of neurons act together to generate an EPSP simultaneously, or you can have temporal, where it's only just where there's only one neuron that generates EPSPs in quick succession. This diagram kind of shows it better. So here you have EPSP and IPSP cancellation, whereas spatial summation is you have two different neurons adding together to reach the threshold, and temporal summation is just that one neuron. This table below shows you some of the excitatory and inhibitory neurotransmitters. I've just written the ones that you need to remember. So glutamate in your CNS is excitatory, whereas GABA and glycine in your CNS is inhibitory. In your peripheral nervous system, it's a little more complicated because acetylcholine and noradrenaline can be excitatory or inhibitory, depending on the type of receptor that they bind to. So depending on the receptor response. Any questions up till here? One. Do graded potentials always stimulate action potential, or can an action potential occur without a graded potential? I'll have to look that one up. I'll get back to you on that one. Would it sometimes take a graded potential of above 35 to stimulate a response due to them dying down? Of above 35? I mean, yeah, because it's dying down, depending on, it's dying down over the span of, like, say, the axon. So say you have a graded potential here, but when it dies down here, when like further away from that point of depolarization, I guess you would need a greater potential at this point to add up above negative 55. Right, okay. What are those? Yeah, EPSPs and IPSPs are graded potentials. So an action potential, yeah, so if, I'll answer that question about action potentials being generated by graded potentials maybe during the break, so I can search it up. But why do we need IPSPs? Why couldn't we just have EPSPs to trigger an action potential? I guess you don't always want an action potential. IPSPs are your inhibitory signals, right? Sometimes you want muscle, say like a nerve terminal acting on a muscle. You want muscle relaxation rather than continuous muscle muscle contraction. And so I guess that's what the action of iPSPs are. It's kind of to balance EPSPs and iPSPs. I hope that makes sense. I just have that one question to get back to in a bit. So synaptic transmission, there's two types. You have electrical and chemical. So your electrical synaptic transmission occurs when current flows through gap junctions, and this is a fast form of transmission that occurs from neuron to neuron, and it allows bidirectional flow of current. So these gap junctions are found where the activity of neighboring neurons need to be highly synchronized. So an example would be your heart muscle cells. It needs to kind of contract in a synchronized manner or simultaneously, and that's where you would find your gap junctions. Your chemical synaptic transmission, on the other hand, it occurs via neurotransmitters, and because of the synaptic cleft, which is a lot larger than the gap junction space, it does take a lot longer to conduct, but it helps amplify a signal, an initial signal, and it can also occur between neurons or from a neuron to a cell. Like the example, a neuron terminating on a muscle at a neuromuscular junction. That's where you might find chemical synapse. So the steps of chemical transmission. First of all, you have your action potential arriving at the axon terminal. One thing to note is that your neurotransmitters aren't synthesized right, like at the point where the action potential arrives. They've already been synthesized previously and packaged into these vesicles, and this is so that you have like faster release and faster rate of transmission. We have a question. So how does a chemical synapse amplify it? Well, because you have a lot of neurotransmitters, and neurotransmitters can act more than once. So it's not just current flows and then it just continues to flow. When you have the release of neurotransmitters, that neurotransmitter has to be degraded first before so that neurotransmitter needs to be degraded first for the response to be stopped. And so that's why, and you also have multiple receptors on the next membrane in which neurotransmitters can act on. And so that's why you can have an amplified response from a chemical transmission. Yeah, no worries. Okay, so once you have the action potential arriving at the axon terminal, your voltage-gated calcium channels open, and this causes your calcium to flow in. The action of calcium in the intracellular fluid is to act as a kind of a intracellular messenger that causes the fusion of these synaptic vesicles with the terminal axon membrane, and causes the exocytosis of the neurotransmitters. The calcium is then removed from the terminal. It's either taken up by mitochondria or actively pumped out by calcium pumps. So the neurotransmitters, once they're in the synaptic cleft, they diffuse across the synaptic cleft and bind onto receptors on the postsynaptic membrane. After they've bound, and this binding causes a response in the postsynaptic membrane. It can cause your EPSPs or iPSPs, and it can cause like another action potential being generated in the next cell as well. Once they've bound, however, they need to be removed, because if you have continuous stimulus from those neurotransmitters, you can have some things like seizures if you've got like continuous stimulus of like muscle cells. So you need those neurotransmitters to be removed. They can be removed in 3 ways. The first being reuptake by and it's an active process, by the presynaptic membrane or by your astrocyte glial cells. You can also have enzyme breakdown. For example, your acetylcholinesterases break down your neurotransmitters. The degradation of neurotransmitters the degraded neurotransmitters can then be transported back into the presynaptic membrane to be resynthesized as well. Or you can just have simple diffusion away from the synaptic cleft. Is chemical transmission unidirectional? Yeah, so you can't really, I don't think you can stimulate the previous membrane, but you can have reuptake by the previous membrane. Actually, you might be able to. Okay, so maybe it's. I believe there's something on. There might be something in pharmacology, but I'm not sure if it's unidirectional or bidirectional action, actually, because it can. There's a possibility it can still stimulate the previous membrane, but I'll come back to that later as well. So the different receptor types that you find at your synapse are either your ionotropic receptors, and these receptors are ion channels. So your ligand gated ion channels, and it allows for really rapid short-lived effects in the perisynaptic membrane. Compared to this, your metabotropic receptors, they are receptors where the receptor and the ion channels are separated, and so you need an intermediate G protein molecule to stimulate the ion channel. Even though it's slower because the response doesn't occur directly, it also typically amplifies the response of an initial neurotransmitter, and that's because you have a lot of intracellular messengers that can act on the on ion channels. It can even act on various ion channels. So I think up to here we have a short break just to stretch, and I'll check the chat for more questions. Are the ions sodium specifically, or just ions in general? So I think you mean this one, the ionotropic receptors. So the receptor ion channels you can allow different types of ions in. So it's not just sodium. You can allow sodium, potassium, and like chlorine ions. I'm sure there's other ions as well. So okay, so just from a quick Google search, yeah, the neurotransmitters can't cause the signal to go backwards. So I think it would be unidirectional. Okay, and I'll get back to that question about the action potentials. What happens when the resting membrane potential? What happens to the resting membrane potential if the sodium-potassium pump is blocked? If it's blocked, I guess you the sodium-potassium pump in relation to your RMP, it helps to maintain the concentration gradient. So perhaps you'd have a decrease in your concentration. That concentration gradient would be less effective when you have when you don't have your sodium being pumped out and your potassium being pumped in. Okay, so we'll keep going on to the next one, which is your divisions of the nervous system. Would RMP be negative 69 millivolts without the sodium potassium pumps? I believe the sodium potassium pumps are what causes the additional negative voltage. So yeah, I think it would be negative 69 millivolts. But your potassium and sodium potassium pumps, they play like a huge, I guess, they're very important in the body. Like they have a lot of roles in the body besides just maintaining the resting membrane potential and returning it back to the resting membrane potential. So if it's blocked, there would be other consequences right, besides just the RMP being negative 69 millivolts. In the lecture slide, it says ANS are located in both CNS and PNS, but isn't the autonomic nervous system just part of the PNS? So the autonomic nervous system, it can have control from, say, your spinal cord, I guess. So here it's just said, it just says that it's part of the peripheral nervous system, but the autonomics do come back to like the spinal cord, which is part of the central nervous system. So your central nerve, so if I go through this first, so the divisions of the nervous system, you start with your CNS, which is your brain and spinal cord. Your PNS is is the communication between CNS and the rest of the body, and that has two divisions into your sensory afferent division and your motor efferent division. Your motor division is further divided into your autonomic nervous system, which controls your involuntary responses, and your somatic ones, somatic nervous system, controls your voluntary movements. Now, like I was saying before, even though it says that autonomic nervous system is kind of part of the peripheral nervous system, it's kind of, because it's like the peripheral nervous system, it communicates between the CNS and the body, and so those nerves from the autonomic nervous system will come back to the spinal cord, which is part of the central nervous system. The ANS, then you have two last divisions, which is your sympathetic division, which mobilizes your body systems in times of stress, whereas your parasympathetic division aims to conserve energy, and so it's your rest and digest responses. We've gone through afferent and efferent terminology. So afferent is sensory, efferent is motor. Ventral means the front side, dorsal, the back side of your body. A ganglion is a group of cell bodies in the PNS, whereas nuclei is the same thing in the central nervous system. Proximal means like closer to the center of your body. Say like your shoulder is proximal to your elbow. Distal is the opposite to that. Ipsilateral means the same side of the body, whereas contralateral is the opposite side of the body. So how is the spinal cord organized? Well, first of all, it arises from the brainstem and ends at around the level of L2, and it travels through the vertebral foramen. So here you can see that there's a vertebral body at the front, and the structures that form your spine at the back, and your spinal cord is a structure that travels through the hole between that. And if you have take a cross section of that spinal cord, you can see sections of white and gray matter. So white matter is where you'll find your spinal tracts, your ascending and descending pathways, and this is where you'll also find your myelinated axons. So those are axons associated with the, the white matter is what you'll find associated with the oligodendrocytes. Gray matter is where you'll find a high aggregation of nuclei and synapses, so where you'll find the cell bodies of motor and interneurons in this space here, and also unmyelinated axons. So if we take a closer look at a cross-section of the spinal cord, and like the peripheral nerves coming out from it or coming into it, you can see in the dark blue is your sensory neurons. They can either come from the back or the front, so either the dorsal ramus or the ventral ramus, but they will always travel in the dorsal root. And you can see their cell bodies are in this kind of ganglion here, which we call the dorsal root ganglion, and the nerve will terminate in the dorsal horn, unless it's a reflex nerve, which will monosynaptic reflex nerve, which we'll get to later, where it synapses here with directly with the motor neuron. In contrast to that, your motor neuron starts at the ventral horn, and they travel in the ventral root, but they can either travel back to innervate, like say muscles in the back or structures in the front through the ventral ramus or the dorsal ramus. I've included here like an annotation. So if you have a cut, say at the dorsal root, you lose your sensory function, but you might lose it. You might lose sensory function in structures in, like, say the skin of the front and the back of that same, innervated by that same spinal cord level. Whereas if you lose motor function, if you cut the ventral root, you lose motor function of, say, the back and the front. But if you, at the point where the nerves start mixing, where you have a combination of your sensory and motor neurons, that's what you call a mixed spinal nerve. So say you have a cut either in the dorsal ramus or the ventral ramus, you would affect both sensory and motor function. And so that's why there's a wider deficit if there's a problem more distal to the CNS. In the gray matter, can we have myelinated axons as well? Because the axons of the end of afferent and the bigger efferent neurons are still in gray matter, and they are myelinated. So can we say gray matter have myelinated axons as well? I think most resources would just say that gray matter will just have your unmyelinated axons. Yeah, most resources would just associate your gray matter with unmyelinated axons, but let me double check. Well, I'll get to that question right after this. Is myelination, perhaps the myelination could be insignificant. So yeah, so that's why maybe the gray matter is not really associated with myelination. So your reflexes are involuntary responses, and it's an instantaneous response to a stimulus, and it's generated at the level of the stimulus, so at the same spinal cord level, and it does not travel up to your brain. The reflex arc consists of your sensory element, so say a sensory receptor, an afferent nerve, your central processing neuron, an efferent nerve, your motor nerve, and then an effector, for example, a muscle cell. Reflex latency or delay is due to like afferent conduction, central afferent conduction delay. You can have delay in the central processing unit or in the efferent conduction as well. And it's affected by how many synapses there are and how many interneurons there are. So monosynaptic reflexes are a direct communication between the sensory and motor neuron in one central synapse at the level of the spinal cord. So in this example here, we have a knee-jerk reflex. What happens is you tap the patellar tendon, and this stretches the quadriceps slightly, and the sensory signal that travels up to the dorsal root ganglion, where you'll find the cell body, and continues into the dorsal horn, but doesn't synapse there. It continues into the ventral horn, where it synapses directly with the motor neuron. The motor neuron then travels and causes muscle contraction, and so that's what a monosynaptic reflex is. Polysynaptic reflexes are a little more complicated, and I've included 2 examples here. And so it's when you have interneurons facilitating the sensory motor communication, and there's more than one central synapse. Polysynaptic reflexes are slower than the monosynaptic ones because there's, again, more interneurons and more synapses. These interneurons, they can, because you have another neuron in between the sensory and motor ones, these interneurons can actually either be stimulatory or excitatory neurons. So in this example on the left, you have touching a hot stimulus, and then it causes the muscle to move your hand away, and so that's kind of like an excitatory response. Whereas this response on the right, it's what you call a Golgi tendon reflex. What happens is you have muscle contraction of the Golgi tendon organ, and the stretch in your muscle fibers causes excitatory signals to travel up to the dorsal horn. Sensory neurons synapse with the interneuron, which synapses with inhibitory motor neuron at the same spinal cord level, and this causes your muscle to relax. One thing to note also is that interneurons, even though the response is involuntary and instantaneous, the interneurons can simultaneously send information of the effector organ to higher central nervous system centers. Where are the okay, so go back because I have a question. So the interneurons in this diagram are in the gray matter. Maybe if I'll use this, the interneurons are here. Okay. So spinal tracts. These are a little bit on. Yeah, they're a little bit confusing to understand, but this diagram you don't need to remember at all. It's just there to be able to visualize what I mean by spinal tracts. So basically what spinal tracts are just areas of the white matter in the spinal cord in which specific ascending or descending pathways travel in. And so they're split up into these kind of sections, and that's what we'll be going through. We'll just be going through some of them. But before that, we'll go through the different types of neurofibers. You have your first order neurons, which are your afferent neurons, and the cell bodies in the DRG. Your second order neurons is the interneurons in the CNS, which synapses with the first order neurons, and it carries like signaling to the thalamus or to the brain. Third order neurons are there if the sensation reaches awareness. So a third order neuron in the thalamus synapses into the sensory cortex. These are the different types of neurofibers that you don't really need to know much about them. It's just because I'll be mentioning things like A alpha, A delta, and this is what I mean by them. They're just different types of neurofibers that have different functions. So say A beta is like mechanoreceptors of the skin, but you don't need to know too much about that. So the first ascending tract is your dorsal column medial lemniscus pathway, or your DCML, and it carries highly localized fine touch by pressure, vibration, and proprioception, but via this A beta fiber. So what proprioception is like knowing where your body is in space. So if I close my eyes and put my hand in front of me, then I know that my hand is in front of me because of proprioception. So how it works is this DCML, the first order neuron, travels on the same side, it's laterally up the spinal cord through the dorsal column, and it synapses at the medulla oblongata, which is the brainstem, with a second order neuron, and it decussates at this point with a third, and then it decussates at the medulla oblongata, and it travels upwards into the thalamus, where it finally synapses with your third order neuron. Your next ascending tract is your spinalthalamic tract, and it provides conscious sensations of poorly localized or crude touch, pressure, pain, and temperature via those A delta or C fibers. And so in this case, your first order neurons enter the spinal cord and synapse immediately onto the second order neurons in the dorsal horn. Your second order neuron decussates in the spinal cord, the second order neuron then ascends contralaterally, so on the opposite side by the spinalthalamic tract. And you can find the third order neurons again in the thalamus, which projects out to the somatosensory cortex. Two questions. Okay, so decussate means to cross over to the other side. In terms of the axon versus dendrite, which part of the afferent neuron is axon and which part is the dendrites? It's pseudo-unipolar neuron. So your dendrites is what it comes out of your cell body. So your dendrites of the afferent neurons would be found in the DRG. Is the primary sensory cortex same as the somatosensory cortex? I think it is, yes. Okay. There's one more ascending tract, which is your spinocerebellar tract for proprioception. You don't need to know too much about this, just that it travels to the cerebellum and it helps with balance and coordination. And about descending tracts, again, you don't need to know too much at all about these. Just know that there's two major groups, your pyramidal tracts and your extrapyramidal tracts. Where the pyramidal tracts, they originate in the cerebral cortex and carries motor fibers to regulate voluntary control of the body and face muscles, whereas your extrapyramidal tracts originates in the brain stem and control your involuntary movements of muscle. Do all ascending pathways enter from the dorsal root ganglion and enter the dorsal horn? Yes, because ascending refers to sensory, so they should all enter from the dorsal root. And do all descending pathways exit via the ventral horn and ventral root? I think they should go from the ventral root, but because descending pathways can be involuntary or voluntary, so I guess some of it can also be your autonomic nervous system. Now, your autonomic nervous system does exit via the ventral root, but there's a different ganglion that you'll learn later that autonomics exit through. Okay, your final slide before the quiz. I don't think we'll have time for the quiz, but feel free to email me if you have questions on the quiz. There's also answers on the quiz as well. It's just like 9 questions, but the autonomic nervous system is comprised of your, again, sympathetic and parasympathetic. So sympathetic is your fight, flight, fright responses, and it acts to mobilize energy. Okay, all good to go up to 10. Okay. So your sympathetic nervous system, it increases blood flow to the skeletal muscles, increases blood pressure and blood sugar levels, and causes pupil dilation. But it decreases blood flow to your gut and skin because you don't need you don't need blood flow to those areas as much during stressful situations. Your parasympathetic, on the other hand, aims to conserve and restore the body's energy, so your rest and digest responses. It's also called your craniosacral divisions. You can sometimes hear it being called your craniosacral division as well, and that's because the parasympathetic nerves are either located in the cranial nerve nuclei or in the sacral region of the spinal cord. It acts to decrease heart rate, decrease blood pressure, but you also have increase of the secretion of salivary glands and digestive glands and blood flow to the gut because for digestion and also contraction of the gut. Does cranial nerve nuclei mean cell bodies of the cranial nerves? Yes, it does. So doesn't the spinal cord end at L2? So how are their sacral nerves? So the spinal cord does end at L2, but you still have nerves that kind of go past L2, like because the spinal cord ends there, but you still have nerves, the peripheral nerves coming out down lower than the spinal cord. I don't have a diagram to show you that here, but yeah, that's how it works. Okay, I'm told I can go till 10:10 for the quiz. So wait, one more question. So if white matter has a spinal tract, how does it synapse? in the dorsal horn, which is the gray matter. So that's, I guess, what it means by the spinal tract is the axons of where it is, but it would need to enter the into the gray matter. But I think no, actually, some you still have some synapses in the white matter, I think. So it doesn't always travel to the gray matter, but it can travel to the gray matter. All right, so we'll start with the quiz. Just answer in the chat, guys, or private message. Which of the following plays a role in maintaining your resting membrane potential? Okay, we've got some answers. It's E, both C and D. And the answer is, yeah, correct. So which phase is characterized by the opening of voltage-gated potassium channels? We've got C, and C is right. So it's not hyperpolarization, because during hyperpolarization you start having the closing of those voltage-gated potassium channels, even though it's low. Next question is, which of the following is incorrect? Got a couple of D's. Yeah, it's D. Which of the following is correct? Yep, we got C, and we've got one question. Wait, why was B incorrect for the previous one? So let me just go back to the previous question. No new action potentials can be initiated during the absolute refractory period. No new action potentials can be initiated during the absolute. Yeah, that's correct. So it's which one is correct? Oh, yeah. Okay. No worries. Yeah. So the answer to number 4 is bidirectional flow can occur through electrical sinuses, which is true. So that's correct. If the nodes between myelin sheath are far apart, how might this affect your action potentials? You've got a couple of D's and E. Okay. Yeah, we've got a couple of these. So the answer is actually A. And this is because these nodes between the myelin sheath are where your ion channels are found. So even if you have, say, the propagation depends on the spread of your sodium, and that's the depolarization from that spread causes the next membrane to depolarize. But if the next membrane, which is at the node, your next ion channel, is far away, then you won't reach there, and so your action potential will stop in its tracks. But I guess if you have myelin sheath, I mean, the spaces between the myelin sheath are not so far apart, but just far apart enough so that the spread of sodium is minimal. I guess it could be D, but I think A would be a better answer for this. Why is it not C? If the nodes between the myelin sheath. Okay, so because even though your myelin sheath help conduct an action potential, you still require, your nodes to be where the action, your nodes are where your ion channels are located, and so that's where you have the exchange of sodium between the extracellular and intracellular fluid. So if you don't have the following like stimulation from the previous membrane, if you don't have ion flow into the next stimulation of the next membrane because it's too far away, then that's why it causes it to stop. But what about unmyelinated axons that can still conduct an action potential? How does myelin sheath actually fasten it? I'm not sure about the technicalities, but I think the explanations I've read is that it just insulates the axon, which somehow makes it faster, the conduction faster. But like you still need like, you know, spaces where the ion channels are. I believe I got this question from, I think it was Khan Academy. So I'll have to review that, and if you guys have any questions, email me, please. Okay. I hope that explains it. So we'll do the last question, and then I think that's all we have time for. Which of the following is true about white matter? Okay, so we got a couple of B's and a couple of C's. So the answer is C, and but it makes sense to think B as well, because you have myelinated axons in the white matter. But it's a trick question because white matter is found in the central nervous system, whereas Schwann cells are cells that myelinate axons in the peripheral nervous system. So the answer is C. They contain ascending and descending pathways. I think that's all we have time for. I'm sorry if I didn't get to answer a lot of your questions, but please email me and I'll try my best to answer them then. Thanks, guys.
Thank you for that. That was really good. And yeah, as she said, the questions will be up as well on the Murus Drive. So if you guys want to look at the last few, you can find them there. So for our next lecture, we have David. And it's also David's birthday, so we wish him a happy birthday today. But yeah, we'll let him take.
Thank you. Where is it? It's over here. Share. Present. So thanks, Sarah. Thank you, Bhavan. Good morning, everyone. I love the voice. I love the speaking. But today we're going to be talking about cancer and hematology. These are just some resources I got some of the information from. Variety of textbooks, lecture notes. I'm in PS Peppa Pig. Shout out. They're better than Bhavan's ones, so go to those. I put some memes up there to make it look like I have a personality. Yeah. Okay, first question. Come on. A, B. Oh no, look at that. You're all wrong. So it's JD from Scrubs. If you said, I had a lot of Good Doctor stuff, which I haven't watched. Yeah, if you say A, I'm concerned about your future as a medical practitioner. But yeah, JD from Scrubs is the best character. Considering I haven't watched the others, but I still know I'm right. So we're going to start by talking about hematopoiesis, which is the creation of cells, blood cells. So what is blood? People mean a lot of things when they talk about blood, and blood has a lot of constituents. So we're going to start by actually defining what we're talking about. Blood can contain red blood cells, white blood cells, or leukocytes. Red blood cells are involved in the distribution of oxygen all over the body, and also the distribution of carbon dioxide, or the removal of carbon dioxide. You have platelets that are involved in clotting. Plasma, which often contains water, proteins, metabolites, nutrients, and plasma And an important differentiation or distinction you should know is between serum and plasma. If you just picture this image, you'll be all right. Serum is plasma without clotting factors. So plasma is in the body. Serum often, it's taken out of the body. The blood will clot because it's removed from the endothelium, and it can be used for blood typing or diagnosis. But serum is just plasma without clotting factors. You can see there's also a buffy coat layer with the plasma red blood cells, and the buffy coat layer contains white blood cells and platelets. First question, what are the requirements? Can you please PM me your answer to the questions on Zoom so everyone has a time to think about it? So direct message me your answers. We'll just get a couple more. Okay, so everyone said D, which is very impressed. And someone said happy birthday to me, so thank you. They got it right, the person said happy birthday. It is D. This is why it's D. It requires iron. This is mainly for the heme unit and hemoglobin, which we'll talk about, and also vitamin B12 and folic acid. And these are required essentially for DNA synthesis. And just an extra thing, does anyone know the antibiotics that inhibit folate synthesis? You can send it to everyone if you want this time. Sulfonamides, beautiful. And what's the other one they give as well? Trimethoprim, exactly. It's sulfonamides and trimethoprim. Not quite methotrexate. But yeah, sulfonatides and maybe methotrexate. But I was thinking sulfonamides and yeah, okay. Next, we're going to talk about where these cells come from. So essentially, they come from stem cells. It's called a hemopoietic stem cell. Hematopoietic hem cell. I definitely said that right. So either what it can do is it can go to a lymphoid or a myeloid progenitor, or it can self-renew. So there's sufficient supply to be continued to grow. If it goes lymphoid, it's going to give rise to your lymphocytes, your T and B lymphocytes, and which will go into your plasma cells. These are important in your adaptive immune system. Natural killer cells are actually important in your innate immune system, so there's a bit of a change there. Myeloid cells are responsible for your innate immune system. You have your granulocytes or polymorphs, which are basophils, neutrophils, eosinophils, and your agranulocytes, which are your monocytes, macrophages, and thrombocytes. It also, erythrocytes are also differentiate from here. PM me again, please. What cell can have both myeloid and lymphoid origin? What bridges them? Yeah, beautiful. Keep going, keep going, keep going. Come on. Yes, more, one more. Good job, dendritic. Everyone got it right. Apologies for my formatting, but I did the best I could. These are, this shows how signaling leads to the differentiation and proliferation of blood cells. Cytokines and protein signalings regulate this, and they're often influenced by the environment. So for example, reducing erythrocytes in the blood, maybe because of bleeding, will lead to significant production of more. So you can see some cytokines like interleukin-3 are abundant. That's generally because these are involved in the proliferation and making a lot more copies, so they're quite abundant, whereas other factors are more specific. Don't need to know much about that. What is the lifespan of red blood cell? PM me. Also, even if you just guess and get it wrong, like I can't describe to you how little I care if you get it wrong, so you might as well just get something. Yeah, everyone got it right, so it's D, 120. What's the main function of erythrocyte? Someone can yell it out, someone can type it to me if they want. You're also allowed to speak. Yep, that's good. O2 transport, oxygen production, really good. Gas transport, so which is essentially the transport of hemoglobin, which carries oxygen to perfuse the tissues around the body. Has other functions, such as facilitating CO2 transport by the carbonic anhydrase enzyme, and it acts as a buffer. Neither of these you need to know. Its morphology, it's often described as a biconcave disc, and it can deform and change shape to squeeze through capillaries. So it's quite malleable in that sense. It's anuclear and has no organelles, which means for energy production, it mainly relies on anaerobic glycolysis and hexose monophase phosphate sometimes. Lifespan is 120 days, as we said. Does hemoglobin itself have greater affinity for oxygen than oxyhemoglobin? Chat, please. Yep, come on, more people, more. Come on, there's 110 of you. I should have 110 answers. I don't care if you're beautiful. Okay. Someone just wrote like one answer 10 times, but yes, it's no. A reduction in affinity of oxyhemoglobin for oxygen will occur with which of the options? Couple more. Okay. Oh, I stop typing for a second. So I want to ask a question. What is the difference between hemoglobin and oxyhemoglobin? Okay, we'll describe that in a second. Keep guessing. So we've got A, B, and D. It's good no one chose C because that was made up. And it's actually B, and we'll discuss why in a second. So what is the difference between hemoglobin and oxyhemoglobin? Oxyhemoglobin has an oxygen bound to one of the heme units. Hemoglobin does not. That's the only difference. One oxygen leads to greater affinity for another oxygen. Important to remember that. This kind of shows the shift of the oxyhemoglobin curve, and it can either right shift will reduce affinity, left shift will increase affinity. And I just define what 2,3-DPG is. It's an intermediate of glycolysis, so it indicates there's a metabolically active drive occurring. And the way that to remember this is think of exercise for right shift. So think of exercise for reduced affinity. When you exercise, you get hot. When you exercise, there's breakdown of metabolism, so you have increased 2,3-DPG. You get increased acidity. A memory tool is I produce lots of CO2. CO2 is acidic, so it's going to increase acidity. That's probably more due to lactic acid production, metabolic functions, but just think you get more acidic when you exercise, more breakdown, more temperature. As unity for hemoglobin and oxygen, which can account for poisoning. Oh, tell me if my internet's not working if I cut out. That's really not good. I changed locations to have better internet. So this is just a description of where the creation of red blood cells occurred during development. In the early weeks of an embryo, it's often produced in the yolk sac. Middle trimester, mainly in the liver, but there's also spleen and lymph node function. And in the last month of gestation and onwards is in bone marrow. What is hemoglobin? There are about 64 million hemoglobins per red blood cell, and there's 4 polypeptide chains in hemoglobin, 2 alpha and 2 beta units. A polypeptide chain and a heme is a hemoglobin chain, and they can loosely and reversibly bind with oxygen. This shows the synthesis. Transferrin is used to transport iron in across the body. Why is the answer to the last question B only? Because alkalosis is going to increase affinity, because when you exercise, you become acidic, not basic. Vitamin B8 are made up, and increased D3 production is going to, oops, my bad. Yeah, that's really bad. Apologies. Yeah, can loosely bind with oxygen. Transferrin is going to transport iron around the body. Ferritin stores it in cytoplasm. You can see how the ferritin leads to iron, which is incorporated into the heme and combines with the alpha and beta chains to create hemoglobin. Iron metabolism, transferrin is transport iron in plasma, and it's often absorbed from the small intestine, whereas ferritin is stored in the cytoplasm. When erythrocytes, hemoglobin is recycled by the monocyte macrophage system, or it's also known as reticular endothelial system. And iron from here that's in the liver. Iron from here is stored as ferritin to be used as acquired. Easy to remember this, your diet contains 10 to 20 milligrams of iron. Oh, this is so bad. I'm so sorry. You're allowed to bully me afterwards if you want. Diet contains 10 to 20 milligrams of iron each day. Divide that by 10 and you absorb 1 to 2 and excrete 1 to 2. This is just diagrammatic representation of ingesting the iron. Only 1 to 2 is absorbed and transferred by iron to where it's required to go. Some of it is stored in ferritin, some of it goes to erythropoiesis and other functions. What is erythropoietin? Erythropoietin is stimulated by the kidneys in order to induce hematopoietic stem cells to kind of proliferate and differentiate. And you want red blood cell production to match red blood cell loss to maintain sufficient perfusion around the body. Increased erythropoietin secretion will increase red blood cell production, and hypoxia is a stimulus for this. So, and that can occur in significant hemorrhage and blood loss as well as exercise. Next question. Oh, where is hemoglobin made? I'll show you. This is in the red cell. Hemoglobin is made in the red cell. I had a question. Yep, question time. Oh, yeah, keep coming. Keep coming. Yep, keep going. Keep going, keep going, keep going, keep going. Go, 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 go. Go, 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 go. Good enough. Everyone got it right, which is ridiculous, by the way. Like, how the hell did you do that? But oh, I didn't even explain it. So when you're at higher altitudes, the partial pressure of oxygen is decreased. That's why it's harder to breathe when you go on mountains and Everest, as you all do. And to compensate for that hypoxia, more erythropoietin is stimulated. I'm so impressed. Every person got that right. That's ridiculous. I wouldn't have gotten that right the first time. Hemoglobin, this is just refers to the breakdown of hemoglobin. I kind of don't know why they teach this to you because you haven't done the liver yet, but hemoglobin is either goes into like a pool, a plasma protein and pool to be recycled. It can go to the bone marrow for the erythropoietin and stimulation of blood vessels, but it's mainly secreted in feces and urine. I think that's all they want you to get out of this because bilirubin and biliverdin are all a bit clinical, but think you maybe know hemoglobin can break down to biliverdin. Erythrocyte abnormalities, there are a lot, and I made it in these two main categories because I don't want to be too scary. It's peer pressure, one person answers and everyone copies. No, because you're all direct, that's why I made you all direct message. We had a good teacher, wink, wink. Oh, that's cute. What do we actually need to know for this? For what? What do we actually need to know for what? For this, it's secreted in feces and urine. That's what I take out of it. I reckon. Yeah, it's recycled plasma proteins traced in bone marrow. Know the word biliverdin, but this is mean. You don't know what the liver does, so how are you supposed to know this? Erythrocyte abnormalities. There's a lot that are specific. Anemia and polycythemia are the two groups I broke it in. Anemia refers to deficiency of hemoglobin in the blood, and this can be due to too few red blood cells or too few hemoglobin in the cells. The answer's on the PowerPoint. That's really funny. Polycythemia refers to an increase in hemoglobin and hematocrit. Or too few. Then how did you all get the Scrubs question? Deficiency can be too few to hemoglobin increase. Okay, I lost my train of thought. I think I explained that. These are, this is from one of your lectures. I don't really get the point, but these are abnormalities. Abnormalities can be in the membrane structure and the antigen-antibody interaction due to an autoimmune complex. In structure itself, which can affect the globin chains, or in sickle cell. What is hematocrit? Hematocrit is, good question. It's involved in the creation of hemoglobin sometimes. Yeah, it's not as important as hemoglobin synthesis, but it's a clinical and diagnostic marker that's more reliable than hemoglobin, because hemoglobin can be altered for a variety of different reasons. Hematocrit is more stable, but you definitely don't need to know what that is in pre-clin, essentially. I've highlighted the ones that are kind of relevant. Again, not so much these two. The production ones, just because of how high yield biochem is, the G6PD deficiency may be looked into. That affects metabolism, can lead to erythrocyte disorders, and hemoglobin production. We talk about these three. This would be, I would ask about this if I wrote an exam question. Other questions? All good. Okay, again, now we're going to do this again, but talk about white blood cells. We've been through this. Lymphoid progenitors, myeloid progenitors lead to myoblasts, lymphoblasts create these cells. Again, colony stimulating factors, interleukins, and other stimuli can stimulate the production of these cells. We talked about what bridges the gap, which was the dendrite. Granulocytes, also known as polymorphs because of their kind of lobular appearance of their nuclei, and agranular, which are lymphocytes and monocytes. What's the difference between a monocyte and a macrophage? You don't need to know. You just need to know what colony stimulating factor is involved in leukopoiesis. Sorry, the question, I should tell you guys what the questions are because I'm not letting you write them in the chat. The question is, which interleukin from the previous page do we need to know? And the answer is, you need to know colony stimulating factors are involved in the differentiation and production of blood cells. Yeah. Monocytes are in the tissue. Yeah, so I asked what's the difference between a monocyte and a macrophage. Someone wrote monocyte on the tissue. Can I have another answer? More answers? Monocytes in blood. Monocytes in circulation, monocytes. Yep, really good. Okay, so actually a little bit of confusion. Monocytes differentiate to macrophages in tissue. So monocytes from the bloodstream differentiate to macrophages in the tissue. This is another example. Polymorph granular. Okay, let's do this. Write in the chat, what is this? Or to me, sorry. Keep coming, go, more. Don't think, just go. Platelets, good. What are these? Oh, thrombocytes, fancy. What are these? Red blood cells, good. What's this? Enlarged nuclei, it's quite bigger than the other ones. We've got lymphocyte and macrophage, monocyte, neutrophil. So guys, remember that our granulocytes are going to have kind of small nuclei that are lobular. This isn't lobular, so it's not going to be a granulocyte. It's quite a big nuclei, which is quite differential for lymphocyte. So yeah, let's see if we have a picture of a monocyte. Okay, I can see why you thought it was a monocyte. Monocytes nuclei are not nearly as large as the lymphocytes. You can see this takes up most of the thing. What's this? Monocyte. Good job. What's this? Getting a bit harder. We've got neutrophil, eosinophil, macrophage. We've already got our monocyte, eosinophil. It is an eosinophil, so then what's this? Neutrophil, more. Good. The reason why this isn't a neutrophil, neutrophils usually have about 2 to 5 lobules, eosinophils have 2. In real life, it's based on staining. You have eosinophil dyes and, what's the last one? Basophil. Yep, basophil dyes. Good job. I'm taking too much time. We're going to run out of time. Oh, God. Okay. So this slide's just saying pinia means reduction, cytosis, and philia means increase. That's, you can just, if you know that, you can figure out the rest of these. Oh, I don't have enough time. Thrombopoiesis refers to the generation of platelets or thrombocytes, and thromboedin is a cytokine involved in this production, and cytoplasmic extensions of megakaryocytes are released in the bloodstream. It's cytoplasmic extensions of the megakaryocyte, which are huge, are released in the bloodstream. This is just showing you clinical effects of platelet number. If you have very low platelet number, you're going to have purpura, which is this bleeding, or spontaneous hemorrhage from mucous membrane. You're going to get a lot of bleeding. And if it's very high, you can also get bleeding because generally they're going to be defective if they're coming out at this amount of time. Okay, now we're going to talk about hemostasis. So I'm about a third of the way through and I have used half my time, but that's not a problem. So there's 4 steps: vasoconstriction, platelet plug, blood clot through coagulation pathway, and fibrous tissue growth to mend hole in the endothelium. Why do you want vasoconstriction to occur? You can call out or you can text me. Reduce blood loss. Yep, good. That's, yep, less blood, good. And you want to limit blood loss so you don't have, you don't bleed out and have shock, and reduce cardiac output and limited perfusion, and then you die. So you want to vasoconstrict to reduce blood through to ruptured valves to produce bleeding, and it also facilitates the platelet plug and coagulation cascade formation. Trauma to vessel wall causes smooth muscles to contract via myogenic spasm. What does myogenic mean? Muscle, yeah, good. Myogenic is a muscle spasm. Autocoid factors, what are autocoid factors? As opposed to endocrine and paracrine. And so autocoid factors are released by the tissue and act on the tissue itself. So it's autocoid factors from intertissues, platelets, and endothelium, such as the reason why does a too high platelet count also cause hemorrhage, because the platelets are probably going to be defective at that high amount, so they're not going to have the appropriate okay. This is this is a platelet structure. It has particular glycoproteins that are necessary, contractile filaments that are required, release of substances to aggregate and activate. So one of these functions are probably going to be inadequately developed if you have too many coming out, because they're not coming out as well prepared. Hope that makes sense. Autocoids such as tissue factor is involved. Which pathway, extrinsic or intrinsic? Very good. So factor 7, both extrinsic and intrinsic. So that's interesting. You're right, often extrinsic and extrinsic will work together, but it is mainly associated with extrinsic because factor 7 is going to combine with tissue factor to kind of initiate the extrinsic pathway, and nervous reflexes. So there's a sympathetic reflex. Platelets release thromboxane A2, which causes vasoconstriction. What drug works by inhibiting thromboxane A2? What's tissue factor? Is that specific? Or I'll tell you in a second what's tissue factor is. Aspirin, brilliant. I didn't think you get that. Really good. This is just showing you how I've oversimplified what I've just said. There's actually a dynamic interaction between platelet activation and vasoconstriction. So platelets can release serotonin and thromboxane A2 once their glycoprotein binds with von Willebrand factors, which we'll talk about in a second, which also lead to vasoconstriction. So it's more dynamic than how I'm telling you in these 1, 2, 3, 4 orders. What are platelets? Due to the turbulent nature of blood flow, many small holes develop in endothelium regularly, and small platelet plugs seal these, and platelets are formed except from megakaryocytes. They have absent nuclei, they cannot reproduce, they have actin, myosin, thrombostenin proteins. So this is so they can contract and heal the hole in the endothelium. They have ER, which store calcium ions, ADP, which is used for activation aggregation of other platelets, and growth factor for healing. So if you have too much platelets, one of these functions can be compromised, which completely impairs the healing process. Von Willebrand factor. Okay, let's explain this. Tissue factor is, it sounds like a very broad name. It refers to a particular autocoid, a particular substance that is released when there's damage to the endothelium that will activate the extrinsic pathway. I have more on that in a second. This here is showing you what von Willebrand factors are. You have platelets with particular glycoproteins that when this endothelium is damaged, it can bind with von Willebrand factors. And these are expressed subendothelially, which means if they can bind with them, that means the endothelium is being damaged. That means they're going to adhere, they're going to change shape, activate, they're going to release granules such as ADP, PAF, and thromboxane A2. We'll go through these in a second. And recruit and aggregation. Spelling. Did I spell something wrong? I'm not sure what spelling is, but yeah, keep going. What do you mean by spelling? Oh, nah, VWF. Ah, von Willebrand factor. I don't quite get how they got that either, but all good. Platelets have a glycoprotein, as I said. They do not adhere on normal endothelium. They adhere to injured areas of endothelium when there's exposed collagen or von Willebrand factors. Exposed collagen is also subendothelial, and they will change shape. They'll activate. So they'll swell, they'll have pseudoprod protrude like hand-like thins, contractile protein contracts, they'll release factors, and it becomes sticky. They secrete ADP and PAF. PAF is platelet activating factor, and release thromboxane A2 to recruit and activate more platelets to all aggregate and come and stick together. And that prevents the blood loss. And it's effective to small traumas, but it's not effective to large traumas. When we have large traumas, we need to stabilize this clot by fibrin crosslinks, forming a more solid plug. And platelets will activate this process often when they're exposed to the subendothelial wall. It's triggered by damaged vessels. It will eventually lead to the, does vasoconstriction or activation occur first? For your exam, vasoconstriction occurs first. In real life, it's more dynamic. The question is, does vasoconstriction or platelet activation occur first? And the answer is, for your exam, vasoconstriction occurs first. But in real life, it's both. Because this is like a really quick process. Like this is seconds, so it's all kind of together. As well, another question that you guys should have asked me is, when someone damages a vessel, when there's injury, isn't there inflammation, and doesn't that cause vasodilation? Does anyone know why it doesn't? Or why it does? Or how can you get a vasoconstriction and also get a vasodilation? So it's a reflex, the vasoconstriction, essentially. This is a really, really quick process. So it's, you vasoconstrict, like when you, someone was telling, like they're a surgeon, when they like stab someone, it's going to constrict for a second, and then they know they have a couple of seconds before blood completely fills out. So you have the vasoconstriction reflex, and then afterwards you'll have your vasodilation. Exactly, beautiful. So that's actually better. So someone said vasoconstriction happens first to try and stop the bleeding, then vasodilation happens later for inflammation and immunity. Yeah, if vasoconstriction occurs, won't the limited number of platelets go into the area inhibiting? So platelets are really small, and this effect, think of it like, so someone asked if vasoconstriction occurs, won't the limited number of platelets inhibit how fast the platelet blood can form? Platelets are really abundant, so they're going to be there. And what you want is, it's more important for, you can think if there's if it's vasodilated, there's much more turbulent blood flow, like there's more flow going. You want to reduce the kind of blood flow to that particular area, so the glycoprotein and the collagen or von Willebrand factors combine together and activate. If it's too quick, like you want it to be as slow as possible. There's platelets are tiny and they're everywhere. Does that make sense? So it doesn't influence how fast the platelet blood can form because the thrombocytes are so abundant. And it's more important that you reduce blood flow to facilitate their formation. I'm going to run out of time. Thrombin catalyzes thrombin to thrombin. Fibrin will enmesh the platelets with the help of factor 13, which cross-links it. And these clots retract. You remember how we had those actin-myosin filaments? These will contract to pull the edges of damaged vessels together. Coagulation cascade. It can consist of enzymes and coenzymes synthesized in the liver, and vitamin K is required for synthesis of factors 2, 7, 9, and 10. What drug works on vitamin K? Quick. Oh, thank you, Bhuvan. Bhuvan said I can go 30 minutes over, which is nice. Warfarin. Can I say your name? Because you're killing it, kid. Yeah. The guy who got the aspirin right got the warfarin right as well, which is really impressive. This is our coagulation pathway. I'm now going to slow down as well because Bhuvan told me not to stress, and I listen to everything he says. This is our coagulation pathway. We can see we were talking about tissue factor before. It's quite specific, and it combines with factor 7 to initiate then the final common pathway. Intrinsic pathway is caused by a damaged surface and will activate. Okay, someone asked me in my PSP how they're going to remember this. And I don't have a good answer for you. All I remember is that extrinsic starts with 7 and common is 10. That means I go 12, 11, 9. So, and I go from 11 to 9 because I know 10 is common and not yet. And I know 9 and 10 is here. So in between, so 9, then 8 is the enzyme that works for 10 to get to the common. I don't know if that helps, but just if you just know it's not 7 and not 10 and 8 is an enzyme, you can figure it out. Because you just go 12, 11, not 10, 9, 8 is an enzyme to 10. And maybe you can think of it, if you have 9 and 10, then this is going to be ascending. So then I need to put 8 in the one where it goes ascending and changes up. 10 to 5, 5 contains calcium. That's not going to help. 10 to 5 divide by, I don't know. Then you just go 10, 5 to 113. Which is like so logical. The reason why they're not named in order is because they were named from when they were discovered rather than their pathway. But you might have to rote learn it. It's really not that bad. And they're not going to ask you. I mean, if they ask you anything more than 7, that would be mean. But maybe you should. I think you should know this. I don't want to tell you the wrong things. Yeah, someone said rote learning works. So maybe hit that up. If you just repeat it enough, you'll do it. But what is extrinsic pathway? It's when tissue factor is expressed by damage in the subendothelial wall, released by trauma. And tissue factor and coagulation factor 7, together with calcium ions, act on 10 to produce activated factor 10. I didn't talk about calcium. Calcium's involved almost everywhere. So in the extrinsic pathway, it's involved here. Especially, it's important in this bit with factor 5, thrombin to thrombin. These stars are all calcium. That should be specified. Yep, to produce activated. So tissue factor and 7 together with calcium ions act on 10 to produce activated factor 10. And the fibrin meshwork leads to the consolidation. And extrinsic is explosive, quicker process than intrinsic, but often they do go together. Do we have to remember which steps require calcium? That's all of them except for this first 2 here. Not hard to remember. So the textbook describes it, all of them except for the first 2 in the intrinsic pathway. Doesn't look like there's any in the cross-linking stage either, but these are activated, so it's not really the same thing for 13. So yeah, just remember the first 2 of the intrinsic doesn't have the rest of it, which doesn't look like it, does it? So maybe there's contention about it, but no, I know calcium is with factor 5, and it's not at the start of the intrinsic. That's That's what I would know. Yep. Intrinsic is triggered by exposure of the blood to collagen or trauma to the blood wall. So subendothelial exposure is more from inside the damage. Extrinsic is more from trauma to tissue itself. Factor 12 may be activated. High is memorizing the intrinsic and pathway, each step important, or just knowing where they occur in the real carcinomas. I don't want to tell you the wrong thing because I don't know what they're going to ask you on exams. I would say do biochemistry instead of memorizing this in terms of high yieldness, but I think it's important. It's good enough to know that extrinsic starts with 7, intrinsic starts with 12. Extrinsic has a tissue factor resulted by trauma. Intrinsic is more by local exposure of collagen or damage to the blood itself, and they have common at 10. No prothrombin, fibrin, and 13 is cross-linking. I don't think you need to know all of this, which isn't as intimidating as you think it is. And the most important is, vitamin K. Which factors does vitamin K contribute to again? That was really quick. Yep, 2, 7, 9, and 10, which are the television channels in Australia that nobody watches. Have I talked about this? I've forgotten where I am. Factor 12 may be activated when it contacts collagen, and platelet activation by damage to the blood wall activates the cascade as well, which is just saying they're dynamic. Yeah, I was confused as well. So someone still doesn't get the difference between intrinsic and extrinsic, which is perfectly reasonable because in practice they're not actually that different. But the way I would do it is intrinsic is exposure of the blood to collagen or trauma to the blood itself. Think of the word collagen when you think of intrinsic. Extrinsic is release of tissue factor caused by trauma. I know if you think of it, if I damage my blood vessel, the platelets are going to be exposed to collagen and there's going to be trauma. Could you say intrinsic is endothelial damage, whereas extrinsic is tissue damage? It's a good memory aid. It's not necessarily true because tissue factor is released by endothelial damage, but it's a good way to think about it. Globally, extrinsic is more damage to the tissue. Endothelial is more scraping. Intrinsic is more scraping. As a memory tool, maybe you can think of it like that. Again, the true answer is intrinsic is triggered by exposure of collagen and trauma to the blood itself. Extrinsic is by trauma that releases tissue factor. Whatever helps you remember that, that's fine. In real life, they're very interconnected and it's very hard to distinguish. The only way you do, the reason why it's important is when you do investigations, you do distinguish. But what do you mean by trauma to the blood? Yeah, so that's a good question. There's platelets in the blood, and I think it's referring to trauma to the platelets. Trauma to the blood is referring to damage to the platelets, I think, in the blood, circulating the blood. I didn't even know what it really meant either, but that's the definition of what it is. But I imagine it had something to do with the circulating platelets being activated by, yeah. I'm sorry, that wasn't very helpful. Trauma to the blood, I think, has to do with the circulating factors and platelets responding to damage within the plasma of the fluid itself rather than by the endothelial wall. And there's a variety of different viruses that can lead to damage to blood itself or to things flowing in the plasma. Yeah. I hope that's okay. It's very complicated and it's actually more than what you need to know what damage to the blood means. But yeah. Have I done this? Okay. Oh, we still have to do cancer, don't we? We've got lots to do. So thrombin cleaves, activated thrombin cleaves fibrinogen to fibrin, and a fibrin polymer is formed by soluble fibrin networks. Use a fibrin creating a fibrin monomer branching into your polymer. This is just showing the importance of thrombin and how it works and all the factors. Just know that it's important. And cross-links occur to create stable clots. Here you can see calcium is involved, but not as important as the potassium. Here is our beautiful prothrombin to thrombin, fibrinogen to fibrin, cross-link fibrin thin, and the release of tissue factor that's activated our extrinsic or intrinsic pathway. Extrinsic, very good. Okay, I'm going to pick it up just to keep engagement as well. Now we need to do stabilization and resorption. So after the clot is formed, it needs to disappear, otherwise you're going to occlude your lumen and you can cause ischemia and necrosis, right? So your fibroblast insert can either insert in connective tissue throughout the clot to stabilize it and to heal the membrane, and contraction of the fibrin plate would stabilize clot. It will resolve by the release of TPA, which stands for tissue plasminogen, and tissue plasminogen activator. And this is, I'll show you in a second, and eventually the breakdown of plasminogen to plasmin. Plasmin will lead to the breakdown of fibrin into, think of D-dimers. It's into several factors, but think of D-dimers. I have more, it's more elucidated in further slides, but D-dimers are measured to see the breakdown. So you can see plasminogen is converted to plasmin. Plasmin is a proteolytic enzyme, means it breaks down protein, it degrades fibrin and some other factors as well. And plasminogen is always trapped in the clot when it's formed, and it takes time to release TPA over a large amount of time and leads to removal of the clot. We're going to talk about D-dimers. D-dimers are a diagnostic tool. If you have very high D-dimer levels, what does that suggest has happened pathologically? Everybody answer. Just guess. Guess more. Go. 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 Just guess random words. I don't care. Go. 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 Keep going. Keep going. Keep going. Kia, write something. Keep going. Go. What does high D-dimer suggest? Nothing happens. Good. Keep guessing. Hemophilia. Keep guessing. Okay. I'm happy with that. High D-dimer means you've had a huge clot, which means you could have had a thrombosis that could have damaged your blood vessels going to organs. It can mean you've had reduced perfusion. It means you're more susceptible to stuff such as heart attacks, and which are called myocardial infarctions, or other sorts of thrombosis in the future. So D-dimers means you've had a huge clot and it's pathological. Thank you everyone for guessing. Coagulation tests, these you should know. Prothrombin time is what? Is it extrinsic or intrinsic? From this image, can you figure it out? The chat has disappeared. Extrinsic and common, beautiful. So prothrombin time is extrinsic and common. Activated partial thrombosin time is then intrinsic and common. Prothrombin time is used to measure the anticoagulant effect of vitamin K as well. And there was a mistake in your lecture slides that I wanted to highlight where they said 12, but it's 7. So keep that in mind. Yeah, thrombin time measures this common pathway from prothrombin. Fibrinogen 11 for deficiencies, and D-dimer for fibrinolysis, which someone got spot on before. Great. What is a thrombosis? A thrombosis is clot formed in circulation. It can be arteries or veins that can block or occlude vessels. An emboli, when fragments of that break off and travel around, and often they're going to get stuck in the lung because the fragment's going to return to the heart. They often form in your deep calves because you don't have valves there. If you're on a plane for a long time, you don't move, you have stasis of your blood, it's going to form a clot that can travel up to your heart, and it's going to pump straight to your lungs, and it can clot your lungs. And then it's it's called pulmonary embolism. It's a medical emergency that can cause ischemia and necrosis, can lead to death. There's something called Virchow's triad, which consists of these 3. I made my own diagram. It consists of endothelial energy or vessel wall dysfunction, which can increase coagulant factors or decrease anticoagulant, changes in blood flow, stasis in veins, turbulence in arterioles commonly, but it can be both, and abnormal coagulation due to changes in components. So hypercoagulability can be a high tendency to form clots, and it can be caused by alterations of coagulation factors. And these 3 factors, endothelial energy, stasis or changes in blood flow, and abnormal coagulation, all lead to thrombosis. It is called Virchow's triad, and it's very clinically relevant. Disorders, primary disorders are of platelet abnormalities or defect in the vessel walls. Coagulation disorders such as hemophilia or factor 8. I'd know hemophilia because it's cool, and von Willebrand disease makes sense. And you can have acquired due to liver disease or vitamin K deficiencies. Oh, sorry, I missed some questions. What is the difference between FGDP and FDP? Good question. Fibrin degradation product is, I don't know. I got to tell you, I don't know. I don't want to lie to you. I imagine that fibrin, I don't know. Good question. Fragments DPs and FDPs. They sound like the same thing, don't they? I'll tell you, there's other combinations of, so like D and E is a fragment X or a Y or something. I don't remember. I didn't put it in just because it's not in your exam, but it's a good question. I don't know. Measure D dimers, and it's a good question. I'm sorry, I don't know the answer to what the difference between FGDP and FDP is. So someone has said FG is fibrinogen degrading product, whereas FDPs is. Okay, thank you, Sriya. So FGDP is fibrinogen degradation product. Thanks very much. Question from earlier, do the 4 hemoglobin units fill with oxygen one after the other? Yeah, I mean, I suppose sometimes it can be spontaneous, but as soon as you've had one, you have a greater affinity for another one. So that's going to happen quicker.
Oh, David, it was just a question that I saw in someone's like practice back, right? It was just like one of the options was the hemoglobin units fill up one after the other when filling with oxygen, as in one first unit fills up, then the second one, then the third, and then the fourth one. But then the lecture notes that I got said, oh, the hemoglobin can cleave itself into 2, and therefore like each like unit of 2 can fill up separately, sort of thing. So I'm not sure who to believe.
So you're not sure if it's sequential or if it's the same time?
Correct. Or randomly, as in like,
yeah,
it can fill up on its own.
I mean, I can guess for you. My friend did biomed, and he told me that he was explaining to me last year that the way it works is there's like an electrochemical change when one oxygen binds, and that has then switches an electrochemical gradient. That means it has a greater like polarity affinity for another oxygen, which would imply it's sequential. I think it's very unlikely. Okay, follow your lecture notes, actually, because I was going to not say your lecture notes, but I reckon it could probably be both. But I would follow your lecture notes, depending on which tissue and what circumstance.
Thank you.
Yeah. The worst thing, I understand when I got the revision lectures, I would hate it when the lecturer would make up like answers. So I really don't want to make up anything. So I don't want to tell you guys something I'm not 100% sure of. The result of that means you can be unsatisfied and I can feel bad. But yeah, I don't want to lie to you. I imagine, I'm going to guess, it's both depending on the situation. I think sometimes you can have one that leads to oxyhemoglobin and then can lead to greater affinity for another one. Or sometimes 2 can come together and are attracted by the affinity and lock immediately. Yeah, I don't know. I'd probably side on sequential, but I don't know. Follow your lecture notes. Yep. We've done that. Okay. These are some hematostatic disorders. Is this, of these 4 options, what do you reckon this is going to be? So we've got a few. Keep going. There's 4 options. Just write one down. Come on. Go. So we've got, it's almost evenly split between purpura and muscle hematomas. I'd say this is muscle hematoma. It's quite significant. It covers quite a large range of area. Purpura is going to be more of these purpley spots, from my understanding. Not worth it. This is just to show you the clinical relevance of how the drugs work. And it's going to take a lot of time. It's not in your exam, so it's not worth it. Can we aim for like 11:15? Okay, we're going to go quick. Warfarin works by inhibiting the liver's production of factors 2, 7, 9, and 10. Cancer. Okay, oh God. Definitions. Will a joint beating just look like inflammation? The reason why I said this isn't a joint is because it's not at a joint. But will it look like inflammation? It depends. It could just also look like bleeding. It could just be blood mass. Okay, I'm going to go quickly now. Definitions: a carcinoma is of epithelial tissue, sarcomas of bone, muscle, and blood vessels, lymphomas of lymph, leukemia of white cells, and it's subclassified according to the tissue they resemble. A squamous cell carcinoma resembles stratified squamous tissue. Adenocarcinomas resemble glands. They can be caused due to damage from DNA that can be inherited or acquired environmentally. And cancer is the evasion of apoptosis, lengthening of telomeres, acceleration through the cell cycle, bypassing checkpoints, diminishing the effectiveness of repair mechanisms, and they secrete hormones that stimulate growth and deactivate evade recognition by the immune system. They can be inherited through sex cell transmission, which are germline defects, and you can tell if you have close relatives with cancer or bilateral impaired organisms. Acquired can be due to a variety of different carcinogens, and these are defects in environmental or somatic cells. Infectious agents such as viruses have been associated with cancer, such as APV and cervical cancer. Smoking is a carcinogen. 90% of lung cancer. Processed meat is in HEP. They tell you that. That's why I put that in. Oncogenes refer to is a gain of function mutation that is dominant and refers and predisposes to the development of cancer, and it's inappropriate acceleration through cell growth. Proto-oncogenes just code for proteins that support growth, division, and adhesion. When they mutate to oncogenes, they become hyperactive. Tumor suppressors are restrained. This is hyperactive, this is restraint. It's genetically recessive and requires both alleles to mutate in the genes. And it's required in checkpoints. You can see some checkpoints involve the adhesion, some checkpoints involve DNA repair, and some are involved in stopping the cell cycle and arresting it. You can see proto-oncogenes, conversion to oncogene leads hyperactivity of protein, not overproduction of protein, and it's actively transcribed in genes. Inheritance, if you're born with susceptibility with an allele being mutated, you can see how it's going to take less time for cancer to develop if it's a two-hit hypothesis. Whereas if you have two protectors, then these are both protective. Sorry, I'm going quickly. Cancer cells often, the development of a healthy cell into a cancer cell is called transformation. It has a lack of contact inhibition, and they do not exit the cell cycle. They just keep cycling through unchecked. They have reduced requirement for growth factors, and they're autocrine for producing their own growth factors, which can act on themselves. They can also lead to angiogenesis and be self-sustaining, escape immune surveillance. They can breach basement membrane by releasing proteolytic factors that make them invasive, and they can be immortal. You can see the karyotype is messed up. You have overproduction of genetic material because of the reduction of repair mechanisms and oncogenic mutations. It's also extension of telomeres, which means they're immortal and they will not die. You'll hear a lot about P53 and RB. No one explained it to me. I want to explain it to you. When DNA is damaged, this is going to trigger factors that will activate P53, that will describe this regulatory protein, which will inhibit this cyclin CDK complex. When it's inhibiting, cell division is blocked, RB is not phosphorylated, and passage does not occur through. If you have mutation of P53, this P21 can't hold these CDK2 and cyclins back, so RB is phosphorylated and it progresses. If RB is mutated, it's phosphorylated and progresses through the cell cycle. What is RB actually? Why is it called retinoblastoma? It's originally a tumor of the retina, and both copies of the RB must be called, which is called a quick answer. What's this called? Both copies must be lost. Two-hit hypothesis. Thank you. It's hereditary, which means it affects both eyes. It's like a paired organ, and a mutant RB allele, chromosome 13, is in every cell of the body. This is a map to remember every type of cancer ever. If it's non-hereditary, usually one eye, one of the paired organs are exposed. Chromosome change only occurs in the tumor cells, and it usually develops at an older age, whereas hereditary is usually at a younger age. RB suppression is involved in the development of many cancers. P53 is associated with 50% of all tumors, and it's involved in the induction of apoptosis and arrest, which inhibits the cell acceleration through the cell cycle, proliferation, and accumulation of errors. You can see the stages. Cancer isn't just cancer happens or it doesn't happen. It requires the gradual acquirement of mutations to lead to the transformation of cancer cells. Usually it requires around 4 to 6 mutations. You can see it's like, think of it kind of as evolution. So you're going to have one advantage that makes it outcompete its neighbors a little bit more. Maybe it can grows proteins a little bit more rapidly. It doesn't repair in errors. And the less it can repair in errors and the more it grows, the more likely it is to acquire additional mutations. You can tell it's a cancer cell because it has an enlarged nucleus and little cytoplasm, and the ratio reports is 1:1 instead of 1:4, and it stains darker because there's excess genetic material being produced. There's a loss of specialized features, which is called anaplasia, or a lack of differentiation. Pleomorphism means it's inconsistent and abnormal. Loss of polarity is essentially, I'm referring to the breach of the basement membrane by proteolytic substances that allow it to grow irregularly, and the evidence of mitosis due to the high rate of proliferation. If it's benign, it's localized and it resembles a sample they came from, and usually not fatal unless it's in a confined space. If it's malignant, it does not resemble the tissue of origin. It's anaplastic, it's invasive, and it can metastasize. And you can see how these are a lot more sinister. The way you can really tell is look for specialized features and the size of the nucleus. You can see this is much smaller than this one. Metastasis refers to spread of the tumor to a site discontinuous with the primary tumor. It can spread lymphatically through lymphatics, through body cavities, through you don't even know what these are, through veins or bloods. You can see so inactivation of checkpoints from repair mechanisms will lead to them being able to do this. They secrete proteolytic enzymes that break through the basal membrane and capillary bed, and they intravascate into the blood vessels. Then you can see they extravascate and colonize over here, and they're going to rapidly proliferate. Intravasculate means
I still have 2 left, so it may as well if I'm not sleeping.
You're not sleeping now. Sorry, Natanya. I'm really sorry. I'm going to mute you. Thanks. Don't be embarrassed, no one cares. Secondary colonization refers to cancer cells which will secrete VEGF signaling of endothelial and grow an outgrowth, and it facilitates nutrient supply and waste removal. It's the cancer cell is going to grow rapidly and outcompete other cells for nutrients, so it needs its own blood supply. They will secrete VEGF to do this, and it facilitates nutrient supply and waste removal. Angiogenesis in more detail, you don't need to know this. You do need to, low yield, you already understand. VEGF stimulate vasodilation. Cells are dismantled, then they're reformed to form new vessels. It's breakdown of basement membrane leads to the creation of new cells. It's not, you get the idea. I think you don't even know the exact specifics. Viruses causing cancer, it's also called transformation. Hepatitis B and C can cause liver cancer, HPV, cervical cancer, and Epstein-Barr virus can cause lymphoma, glandular fever. Contact inhibition, contact inhibition is more referring to a petri dish. When they have cancer cells, the normal cells don't grow beyond, like they don't stack on top of each other. Once they reach the edges, they no longer continue to grow. Cancer cells don't have that sort of inhibition, so they're not influenced by the cells around them. They continue to proliferate. The question was, what is contact inhibition? I hope that makes sense. I should probably say the question before I answer the question. Why is cancer bad? Okay, how much time do I have? How quick do I need to go? Oh, I got time. Cancer is bad because it displaces normal tissue that compromises function, particularly when anaplastic or non-differentiated cells replace specialized cells. It can block or occlude partially or entirely occlude lumen of the bowel, arterial, venous, or lymphatic supplies, which can have deadly consequences. And because it's rapidly proliferating, whereas normal cells are not, outcompete normal cells for nutrients and blood supplies, which means the organs and tissue often undergo catabolism themselves or break down to feed the high metabolic demand of cancer, which is responsible for the clinical sign of cachexia or weight loss. Why does cancer come back? It may result from a failure of cancer to develop blood vessels, a lack of angiogenesis initially, and resurgence maybe to a new means of acquisition of blood supply through maybe through angiogenesis or whatever other mechanism. Genetic predisposition, some cancers are quite common. 1 in 10 women get breast cancer, and penetrance refers to the percent of individuals with genetic defects who will acquire the phenotype of the disease. So you might have the allele or the mutation for colon cancer. Almost everyone who has it gets colon cancer, whereas breast cancer, you might have the BRCA gene mutations, but not everyone who has that develops breast cancer. And this is showing you how p53 inducts apoptosis, and usually leads to, when repair fails, usually leads to the cells control it. Mismatch repair genes are upstream to p53 often, and they repair DNA if it's damaged due to stress or mutation, and usually occurs in the S phase, has a 2-hit hypothesis, and has loss of function. Familial adenopolyposis is autosomal dominant. It has a penetrance of about 100%. By the age of 16, adenoma usually develops. By 39, colorectal cancer usually develops. These are benign polyps, but as you can see, eventually they're going to develop to become cancer, very susceptible. They also have malabsorption, so it's very unpleasant disease for people who have it. You need about greater than 100 required for diagnosis, but you're going to check them continually. Breast cancer is up to 10% due to genetic predisposition, and BRCA affects breast and ovaries. So if you have a history of ovary and breast cancer, that's very suggestive. BRCA1 and BRCA2 are susceptible to these genes, and your increased likelihood if you have a family history of early age onset of breast cancer, breast and ovarian cancer family history, Ashkenazi Jewish, which is risk factor for everything, and female and a family history of male breast cancer as well. You must also remember male breast cancer exists. Chronic myeloid leukemia, this is disgusting. I'm sorry about my formatting, but it refers to the translocation of chromosome 9 and 22. What does that mean? So why is it bad? Chronic myeloid leukemia is a blast crisis, which can be of either myeloid or lymphoid origin, even though it says myeloid here. That's not particularly true. It can be myeloid or lymphoid origin. More of it is myeloid. and it probably originates due to pluripotence, mutation in pluripotent stem cells. Tyrosine kinase receptors are two sort of receptors that when they dimerize or come together, they lead to the phosphorylation often of like G substances or activate CAMP. They need phosphorylation. They phosphorylate substances which have a downstream effect which promote growth and protein production. CML is due to a 922 translocation. The tyrosine kinase is on chromosome 9. That's the receptor that leads to the proliferation of protein. BCR is on chromosome 22. This is a Philadelphia chromosome, which is responsible for the ABL tyrosine kinase. Fusing to BCR ABL tyrosine kinase, it becomes overactive, rapidly promoting proliferation and cell growth by bypassing, inhibiting, and evading apoptosis. As you can remember, the dimerization is constant. It's constantly phosphorylating, leading constant protein production, as constant oncogenic activity, and tyrosine kinase receptors are quite implicated in many cancers. Oh, it's my last slide. I finished on time. I finished 5 minutes early. I shouldn't have skipped all the questions. So what treatment is used for this? Do you remember? It's our last question, so everyone has to write. I will have 110 answers. 115 answers. I don't remember is a valid answer as well. I need 115 I don't remember. Come on, more people. No, I don't know. Good. Happy birthday is definitely valid. Keep going. More. I don't know. I don't know. I never knew in the first place. Not too sure. I don't know. Happy birthday. Thank you. Thank you. Thank you. Okay. You're all being very kind to me. The answer is Gleevec. And Gleevec works by inhibiting dimerized BCR ABL tyrosine complex by binding to the ATP binding site, which inhibits downstream activation. So if you look at this image, usually you have ATP is binding here, leading to the proliferation of protein and this cyclic AMP downstream signaling. This is an amazing sort of drug because it's specific for the specific BCR ABL tyrosine kinase, which is insane. So it's specific for only the tyrosine kinase or this mutation, and it inhibits ATP binding and the downstream proliferative effect. So the ATP pocket is unique among tyrosine kinase and doesn't have significant effect on others. Potassium is required for potassium is required for what? Yep, you got it right. Good job for D. 13. It's 2, 7, 9, and 10. I think I meant to make 1, 2. I did this last night. Well, not last night, I did it before, but still. The penetrance of colonic adenomous polyposis colon cancer is 100%. What happened here? Oh, good. Cancer cell histology is characterized by. Yes, good for all the responses. Cells explain. You're right. Why do I have potassium? What was I thinking? What was I thinking? I think I wrote that question for potassium, but vitamin K is what you need to know. So ignore the potassium. You don't need to know about potassium. Are all the answers D? Maybe. Which of these viruses may cause cancer? More. Last question. Keep going. We've got until 11:15. We have 2 minutes. Keep going. Epstein-Barr virus can cause glandular fever, which is associated with Burkitt's lymphoma. I guess we'll never know what I was thinking. Sometimes I'm so brilliant, I don't even know what I'm doing. Are there any questions at all about anything related to med, about your exams, about medicine, about how to study? Any questions? Happy to answer. Hit me up on Facebook if you want. You can email me. I'm sorry for a couple of mistakes. I'm sorry I went quickly, but yeah, ran out of time. Yeah, you'll do fine. Study biochem. You'll be fine. Yeah. Thanks so much, Bhuvan. Thank you, everyone.
Thank you, David, and happy birthday as well. A lot of commitment coming on your birthday, but that was a great lecture, so thank you so much. And sorry we had to rush you at the end.
No, it's necessary.
Anyways, I thought before Pharm, since there's 2 more lectures, we'll give everyone like a 5-minute break, just to like relax, maybe go to the toilet or whatever, grab a drink. And so we'll like start back at 11:20 for Pharm, if that sounds good with everyone.
Have a good day, everyone. Goodbye. Cheers, Bhavon.
Anyways, thanks, David. Okay, yeah. So I guess we'll start with Pharm, and welcome to Julia, who's going to take us for this lecture.
Thanks, Bhavan. I guess I'll just get started now. Thanks everyone for turning up and sticking through the first couple hours. So well done. Unfortunately, I will have to go quite quickly as well. Normally you'd have 2 pharmacology revision lectures this semester, but as I understand it, you didn't have the one in semester, so I'll be going over everything. But yeah, if you have any questions, I don't have the setup, like a 2-screen setup where I can see the chat. So I can answer any questions that you have after the presentation and at the end if we have time. And you can always email me or message me on Facebook about anything as well. So yeah, I'm Julia, and I just want to start by acknowledging the traditional owners of the land on which I'm presenting the presentation today, and pay my respects to elders past, present and emerging, and also to any First Nations people present today or watching the recording as well. So this is kind of a brief overview of everything I'll try and touch on today. As I said, I do have to go through everything, and I know I'm not going to cover everything in a great amount of detail, but hopefully the aim of this is just to trigger your memory, to test yourself if you feel comfortable on a topic or if you need to do a bit more revision. So I'll be asking some questions as we go. You can either unmute and shout it out, but I probably won't wait for an answer all the time because I know it can be like a bit intimidating. So it's just an opportunity for you to think of the answers in your head and test yourself along the way. So it's kind of the goal today. So start off with a few questions for you to think about. So what is the pharmaceutical phase? What is the pharmacokinetic phase? And what is the pharmacodynamic phase? Again, you can put in the chat if you want, but I can't see it. So right, actually, let me try and get it up. Okay, moving on. So the pharmaceutical phase is the drug being released from its package. The pharmacokinetic phase, as I'm sure you've heard, is what the body does to the drug, and the pharmacodynamic phase is what the drug does to the body. So the aspects of pharmacokinetics that we're going to look at today is administration, absorption, distribution, metabolism, and excretion. And the aspects of pharmacodynamics that we're going to look at as well are the receptors or the drug targets, which are not always receptors, the drug binding, and the reaction pathway. And what I mean by the reaction pathway is how does the drug binding to its target actually cause a response? Another name that you might hear for the pharmacodynamic phase is the biophase. Okay, so looking at administration of drugs, what does bioavailability mean? Great, so bioavailability is the proportion of the active drug that reaches the systemic circulation. Here are a couple more questions that you can think about, and then I'll share the answers too. So which route of administration would be the best for patient compliance? What does parenteral administration refer to? In what situations would you avoid administering drugs PO or by mouth? And which route of administration skip the first pass metabolism at the liver and therefore increase bioavailability? So for the answers. The oral route of administration is really the best for patient compliance. It's very easy for patients to take it, and they can self-administer it rather than going into a center and having to get an IV drip, for example. So parenteral administration refers to anything other than oral. So injection, inhalation, topical cream, there are many more. If the patient is vomiting or unconscious, it's really impractical to administer a drug by mouth. Also, if you want a drug to have a very localized effect, you might not want to administer it by mouth. For example, like a topical cream like Deep Heat, you would just want it on the site that you might be feeling pain rather than kind of a systemic effect. Also, if the drug has poor oral bioavailability, so if a lot is deactivated in first pass metabolism, you might want to consider another route. And kind of moving on from that, IV administration has 100% bioavailability, whereas in oral administration, as I said, we have first pass metabolism at the liver. So that deactivates some of the drug before it reaches the systemic circulation, before it even has a chance to exert its effect on the target. Okay, so we have to consider 3 key factors when we're talking about the absorption of a drug. So the first factor on the left is the size. So if something's smaller, it's more readily absorbed. The second factor is polarity. So this comes into play when we're talking about the absorption of drug, but also about side effects as well. So if something is non-polar, as we know, that means it is lipophilic. It can cross the phospholipid bilayer, and therefore it can get into the cell, into all our cells. So it might have potentially more side effects. Whereas if something is hydrophilic, it's water soluble. It's much more likely to stay in the bloodstream rather than diffusing out and entering all the tissues. The third factor, which is, I've underlined it because it's definitely more important than size, and it kind of, it's pretty important, is ionization. And so the ionization of a drug depends on 2 factors, and that is the pH of the compartment where it's in, so like the stomach, which is acidic, or the bloodstream, which is not so acidic, and the pKa value of the drug. So personally, I found this, a bit more confusing, especially compared to size and polarity. So I was just going to go over this a bit to hopefully clarify it or reiterate it. So most drugs are either weak acids or weak bases. And when we talk about the pKa of a drug, we're talking about the pH at which 50% of the drug is ionized and 50% is neutral. This is an important question to consider, is which form of the drug can actually be absorbed? Is it the ionized form or the unionized form? I've actually got the chat up, so feel free to send some answers through. And if you're not sure, that's okay. Great. Everyone's pretty on top of this. So the unionized form can be absorbed, which is a really key message. So the takeaway here is that a weak base is better absorbed in a basic environment, whereas a weak acid is better absorbed in an acidic environment. So if we apply this to the weak acid example, if we have an acidic environment, we have lots of H+ ions on the left. So that's going to promote the reverse reaction here, producing AH, the unionized form, which is more likely to be absorbed. So again, weak base is better absorbed in a basic environment, whereas a weak acid is better absorbed in an acidic environment. So hopefully that made sense to you. If not, I'm sorry. Okay, so I'm sure you've seen this graph before as well. So some key terms to know here. MTC is the minimum toxic concentration. So this is the line between the green and the red. And this is the minimum dose of the drug at which you get an adverse effect. The MEC, on the other hand, is the line between the yellow and the green, and that's a minimum effective concentration. So the minimum dose of the drug required to produce, a desired response or have some kind of desired effect. The therapeutic index is the ratio between MTC and MEC, so the toxic concentration and the effective concentration. It's important to get this one the right way around. So yeah, remember MTC is divided by MEC. So what is the significance of a low therapeutic index? So if we have a low therapeutic index, that means our minimum toxic dose and our minimum effective dose, they're very close in range. So it's very risky to administer this drug, because if we don't get the dosage exactly right, there's a high risk of adverse effects. Yeah, okay, so the next pharmacokinetic factor that we're going to discuss is distribution. And the concept that you've learned about here is volume of distribution, which is the dose divided by the plasma concentration of the drug. Initially, that might not mean much, but when we talk about high volume of distribution and we look back at this equation, if we have a high volume of distribution, that means we're going to have a low plasma concentration of the drug. That means there's not that much drug in the plasma compared to the dose that we've given. So it's all being absorbed out of the bloodstream and into the tissues. So as I briefly mentioned before, some characteristics of this drug that might have a high volume of distribution, it might be small, non-polar, and uncharged. So it can really readily be absorbed into the cells. However, if we have a low volume of distribution, looking back at this equation again, that means we have a high plasma concentration of the drug. Not much of the drug is actually leaving the plasma at all. So that means it's more likely to be bound to plasma proteins. Next, metabolism and excretion. So we've briefly touched on first pass metabolism at the liver. And importantly, the liver uses a class of enzymes that you'll see all the time. So the CYP450 class. And the point of that is to make drugs more water soluble for the kidneys to excrete. So lipid soluble drugs will become more water soluble at the liver, and then they can be excreted by the kidneys. However, there are other, although less common, routes that drugs can be excreted. So what are some of those other options? Yep, I've got skin, lungs, and so by skin it can be sweat. We can also have things like saliva and even breast milk. Okay, so this, yeah, I missed a slide. So in terms of excretion, we have two main, I guess, mechanisms, the first order kinetics and the zero order kinetics. First order kinetics is what happens when you increase the concentration of a drug, you also increase the rate of elimination. So the enzymes, the liver enzymes, the CYP450, they haven't been saturated yet. So as we add more, the liver can kind of cope with that and accordingly increase the rate of elimination. So it's pretty good. However, after a certain point, if we keep increasing the concentration of the drug, we can get to zero order kinetics. So the liver enzymes are saturated and the rate of elimination is constant. So even though we're adding more drug, we can't eliminate it as quickly and the drug builds up in the circulation. So important example of that is, of course, alcohol and aspirin as well. This is a diagram of this mechanism. I find diagrams are a really good way of explaining this concept. So you can definitely look at that more in your own time as well. Okay, so now we've touched on pharmacodynamics. So what the drug is doing to the body. So the first thing we're going to talk about is drug targets. So what are some general examples of drug targets? Talked about receptors, but there are many other things that drugs can bind to and not just receptors. So some of these other targets could be enzymes, they could be, I guess, carrier proteins or transport proteins, really anything, not just receptors is the main point that I wanted to get across. Okay, so the next thing is selectivity versus specificity. These words sound really similar and it can be a bit of a confusing topic. So I've made a bit of a diagram on the next slide to explain this. So imagine we have a drug that's trying to inhibit a target enzyme. And this enzyme happens to be found in the brain, the heart, the liver, and the hands. So the drug wants to, block this enzyme, but unfortunately, it's only a selective drug. So the drug binds to the enzyme wherever it's found. So even though the drug might only want to target the heart, it's going to bind to the enzyme in the heart, but also in the brain, liver, and hands, just wherever it is. However, if this cardio drug was specific, even though this enzyme that it's targeting is located everywhere else, somehow it would be able to identify and only act on those enzymes in the heart. Unfortunately, most of the drugs that we have are only selective and not specific. So they'll just bind to the enzyme wherever it's found, and it's hard for it to distinguish between the same enzyme in different locations. Okay, the next factors that affect drug binding are affinity and efficacy. So affinity is the strength of binding. So I think of affinity, attraction. So, and whereas efficacy is the ability of a drug to cause a response. And so we can use these terms in relation to the types of interactions, so agonists versus antagonists. Agonists have high affinity, they're able to bind to the target site, and they also have high efficacy. agonists cause a response. On the other hand, antagonists have high affinity. They're able to bind to the target site, but they have no efficacy because antagonists want to block the action of that enzyme or receptor or anything or something like that. So antagonists have high affinity, but no efficacy. They don't want to cause a response. We also have partial agonists, which honestly you don't really see that much of. It's pretty much just an agonist, but not as good and kind of less effective. So yeah, don't worry too much about that one. Okay, now we're moving on to some, I guess, nervous system stuff and then neuromuscular blocking drugs as well. So the top diagram represents a cholinergic nerve. So we can see we have a long presynaptic neuron, and at the synapse we have acetylcholine communicating with the nicotinic receptor. And then in the postsynaptic neuron, we have acetylcholine binding to the muscarinic receptor. And this is a parasympathetic nerve because of the acetylcholine binding to the muscarinic receptor in the postganglionic neuron. So what is the effect of the parasympathetic nervous system? So what kind of responses are we going to get from acetylcholine binding to the M receptor? Yep, seen a lot of good words in the chat. So our muscarinic receptors, key term, rest and digest, and then you can work backwards from there. So we have parasympathetic nerves in the GIT, lungs, blood vessels, and as part of the rest and digest, we're going to promote digestion. We're going to vasodilate the vessels in the GIT and the organs. We're going to constrict our pupils because we don't need supervision when we're just kind of chilling out. We're going to slow the heart rate and decrease cardiac output. We're also going to bronchoconstrict as well. So the second example here, we have acetylcholine. We only have one neuron from the CNS. So this is somatic nerve. And so this is a nicotinic receptor that's going to act at the skeletal muscle at the neuromuscular junction. Another important thing to note is that we don't just have nicotinic receptors at the NMJ. We also have nicotinic receptors, as you can see in the top diagram, at the ganglia between presynaptic and postsynaptic neurons. Okay, so the bottom example is of an adrenergic nerve. So the presynaptic neuron is the same, although the presynaptic neuron is shorter compared to parasympathetic. However, in the postsynaptic neuron, we have noradrenaline. So what types of adrenergic receptors might noradrenaline bind to, and what kind of response would that have? Okay, yep, so adrenergic receptors, so we have two main classes, alpha and beta, and that's going to be the opposite of our rest and digest. It's going to be our fight and flight. So increasing our heart rate and cardiac output, bronchodilating, dilating our pupils, stopping our digestion, dilating the blood vessels in our muscles, and yep, a bunch of other fight and flight effects. So the alpha receptors, the way I like to think of it is that the beta receptors are in the heart and lungs, and the alpha receptors are pretty much just everywhere else. So the two types of beta receptors that we have are beta 1 in the heart because we have one heart, and beta 2 receptors in the lungs because we have two lungs. And then everywhere else has alpha receptors. Okay, so it's all, good to have our noradrenaline acting at, our target site. But how's it actually deactivated? We only want a very brief response to kind of prevent continual stimulation of the postsynaptic, neuron or the target organ. So the two ways that noradrenaline is actually deactivated is that it can be reuptaken into the presynaptic neuron to kind of remove it from the synapse and stop it acting on the alpha and beta receptors. And the second mechanism is that we have enzymes here in the synaptic cleft to break down the adrenaline or noradrenaline. And the NE in this diagram just refers to norepinephrine, so kind of the American term. And so the two enzymes that can break down the neurotransmitters, or the adrenergic neurotransmitters, are monoamine oxidase and catechol-O methyltransferase. So I guess the takeaway message is that we do have to have some way of removing these neurotransmitters, and it can be reuptake or by enzymes. Okay, so what about acetylcholine at the neuromuscular junction? How is that going to be deactivated? So this little thing here, it's a bit pixelated in this diagram. We have acetylcholinesterase. So it's an enzyme that's going to break down the acetylcholine in the neuromuscular junction and then prevent it from binding to the nicotinic receptors. So the core drug that relates to this topic is pyridostigmine. So this is a reversible anticholinesterase. So it's going to stop anticholinesterase. Therefore, there's nothing to break down the acetylcholine at the neuromuscular junction. It can keep activating those nicotinic receptors and causing a muscle contraction. So what side effects might this have? Yep, so we're increasing the concentration of acetylcholine, and this can act at muscarinic receptors as well. So we're going to have an increase in our parasympathetic nervous system activity, increased rest and digest effects. And also the context in which this is used is myasthenia gravis, which we'll discuss later as well. Ooh, okay. Okay. This is a little Who Am I game I thought might be fun to test your knowledge of the neuromuscular blocking drugs. So I am a competitive antagonist at the nicotinic receptors at the neuromuscular junction. I cause muscle paralysis by stopping acetylcholine from binding to the nicotinic receptors. However, I can be overcome by increasing the concentration of acetylcholine, such as by prescribing pyridostigmine. I'm a non-depolarizing blocker, and my name is rocuronium. So it's a competitive antagonist, non-depolarizing blocker. Second blocking drug. I'm a competitive agonist, which overstimulates and permanently depolarizes the nicotinic receptors. After an initial muscle twitch, I also cause paralysis. I'm a depolarizing blocker, and my name is suxamethonium. So good job to anyone who was able to guess that. Okay, so the next core drug is atropine, and it is an anticholinergic drug. And so its mechanism of action is blocking the muscarinic receptor. So it's a muscarinic antagonist. So it prevents acetylcholine from binding to the M receptors. So we're inhibiting the parasympathetic nervous system. So the side effects will relate to sympathetic nervous system activation. So what are some of examples of the side effects? So there are a couple of ways to remember the side effects of sympathetic nervous system activation. I just prefer personally to remember it as fight or flight and then kind of work backwards from there as to what effects that would have. So dry mouth, constipation because you're stopping digestion, bronchodilation, pupil dilation, and skeletal muscle vasodilation. However, another acronym that kind of goes around is like the days of the week. So Monday, Tuesday, Wednesday, Thursday, Friday. And so that kind of stands for muscle cramps, twitching, weakness, tachycardia, so increased heart rate, and fasciculation, muscle twitching. Personally, I don't find this one as useful because I feel like it doesn't go into the scope of side effects that, do happen in the sympathetic nervous system, but if it works for you, it can be good. And the last condition we'll talk about here is myasthenia gravis. So this is an autoimmune disease, so where autoantibodies, recognize or mistakenly recognize the nicotinic receptors as non-self and then attack them at the NMJ. So what kind of effect would this have on a patient with myasthenia gravis? Yep, so there's fewer receptors for acetylcholine to bind to. So you get fewer action potentials. So you might have weak and unsustained muscle contractions and the person would fatigue very quickly. However, they actually do kind of recover their energy after a short period of rest. And the drugs that we can use, which we've briefly discussed just before, are pyridostigmine. So this is an anticholinesterase. As I mentioned before, we're increasing the amount of acetylcholine, so there's more chance of a muscle contraction. However, the increasing acetylcholine will act at the nicotinic and the muscarinic receptors. So we want to use atropine to block the muscarinic receptors. So the acetylcholine can only act at the nicotinic receptors and we don't get all those parasympathetic nervous system side effects. We also might want to limit the autoimmune response by prescribing a corticosteroid or another immunosuppressant, but don't worry too much about that. Okay, so now we'll move on to antibiotics, and this is a pretty hefty part of the course. I'm just going to cover the core drugs, and the main thing, the main structure I'm going to use is kind of identifying the drug target, the class, whether it's bacteriostatic, bactericidal, and then other details like specific adverse effects. And then if you personally really want to go into detail, you can look at the spectrum and other things like that. But this is a good way of structuring revision and learning around antibiotics, I find. So I'm sure you've seen, e-pharmacology left, right, and center, but I really do recommend e-pharmacology, especially if you're looking for more information on this presentation today. It has a lot of detail, but it also identifies the core drugs that you need to know, not just for these exams, but throughout the next few years. So, yep, good to get on top of that. Before we go into the antibiotics, I'll just mention quickly the antimicrobial creed. This does come up on questions, even though it might not have been mentioned too much in class. So I would recommend having a quick review of this, and it also is really important in preventing antibiotic resistance as well. The only thing that Monash usually changes from the last point is the last E, where it says ensure monotherapy in most cases. Monash likes to kind of teach it as ensure like oral therapy in most cases, just due to increasing patient compliance. Okay, so to start off with, I'll give you a few moments to think of which of these antibiotics will inhibit cell wall synthesis. There are multiple on this list. So I'll give you a few moments for that. Yep, so we have amoxicillin, isoniazid, which is a bit of an exception because it's one of the ripe drugs. I'll go over that. And we also have cephalexin and vancomycin. Just to note that all drugs that inhibit the cell wall are bactericidal. If the bacteria doesn't have a cell wall, it will die immediately. Maybe not immediately, but it will die. Anyway, so we'll start off with amoxicillin, and this is by far the most common antibiotic, so you'll really see this around a lot. Its class is beta-lactam, and more specifically penicillin. As I said, it inhibits cell wall synthesis and does this by inhibiting the transpeptidase enzyme, so inhibits the formation of crosslinks between the peptidoglycan molecules in the cell wall. Other details, it is a first-line antibiotic, as I said, used a lot, broad spectrum, and it's really good for gram-positive infections. And I'll go over as to why in the next slide. There is quite a risk of allergy and also of superinfection, which is when you're killing all the natural flora from prescribing an antibiotic, so you can get a secondary infection because you don't have those natural flora as part of the first-line defenses. You might see amoxicillin in the context of augmentin, where it's combined with clavulanic acid. So some resistant bacteria have developed an enzyme called beta-lactamases. So they actually can break down amoxicillin or other penicillins and make them ineffective. So we prescribe clavulanic acid to stop the beta-lactamases in the bacteria and therefore increase the efficacy of amoxicillin. The next drug, cephalexin, is very similar to amoxicillin. It is also beta-lactam, but more specifically, it's a cephalosporin. And as you can see, it inhibits transpeptase enzyme as well. If patients are allergic to penicillins, would you give them cephalexin? It's a really important question to consider. So keep the answers coming in the chat. Yep, seeing some mixed answers. So the answer is no. As you can see, they have, like, you know, they target the same enzyme in their mechanism of action. So if a person is allergic to penicillins, they could possibly have an allergic reaction to cephalexin or cephalosporins as well. And when a patient's already sick, this is the last thing you would really want. So don't risk it, is the key message here. Okay, so this is a diagram just kind of explaining why I said that amoxicillin and other cell wall inhibitors are particularly effective on gram-positive bacteria. So this inhibits the peptidoglycan cell wall, and we see in gram-positive, they have a really thick cell wall and it's not under this outer membrane. So that's why cell wall inhibitors are particularly effective for gram-positive infections. The next drug is vancomycin, and so its class is a glycopeptide. It's a transglyosidase inhibitor. So this is just another enzyme that is responsible for kind of building that peptidoglycan cell wall. I guess in contrast to the amoxicillin and cephalosporin, which are pretty, you know, first-line therapies, this is a really last-line therapy for severe gram-positive infections, such as MRSA, so methicillin-resistant Staph aureus, or C. diff. Has quite distinct adverse effects, which can come up as kind of buzzwords on exam questions. And this is red man syndrome. So that's pretty much exactly what it sounds. So mass vasodilation and histamine release caused by vancomycin, causing a red appearance. It's also toxic to the kidneys and the ear. You know, funny memory tool that has been floating around the last couple of years is a red man called Mr. SA driving a van. So it sounds pretty bizarre, but it actually includes a good adverse effect. So red man syndrome called Mr. SA, so referring to MRSA or severe gram-positive infections, and driving a van, so vancomycin. So if that works for you, then that's good. And the last drug that inhibits the, I guess, cell wall, it's not really a cell wall, but it's technically in this class that I'll touch on, is isoniazid. So this is an antimycobacterial, so effective for any mycobacterium infections such as tuberculosis. So it inhibits the mycolic acid synthesis. So we can see mycolic acid is this kind of, you know, waxy lipid layer that is above the peptidoglycan cell wall in Mycobacterium. So the normal cell wall inhibitors aren't really going to be effective. We need isoniazid. So this is, again, part of the RAP drugs. And all of these RAP drugs, including isoniazid, are hepatotoxic, so toxic to the liver. Okay, so which of these antibiotics inhibit protein synthesis? Yep, so we have doxycycline, gentamicin, erythromycin, clarithromycin, and chloramphenicol. And I'm just going to go over these quickly as well. So doxycycline is a tetracycline antibiotic. The class is pretty easy to remember because it has the same ending. It inhibits protein synthesis, more specifically by binding reversibly to the 30S subunit of the ribosome. It's bacteriostatic, so it only stops the growth rather than actually killing the bacteria. This means that it relies on the host immune system to actually kill the bacteria. It's pretty broad spectrum as well. And importantly, it's not only used for bacterial infections, it can also be used to treat acne in really severe cases, and also as malaria prophylaxis. So this can also come up in exam questions, and it's pretty unique in this sense. It also has pretty unique adverse effects. So it depletes the calcium stores in your body. So that can lead to yellow teeth and also impaired bone growth. And this is why doxycycline is contraindicated in children under 8 years old and pregnant women, because these are two kind of groups of people that we wouldn't want bone growth to be affected in. It can also cause photosensitivity. So sensitivity to light mean that people can get sunburnt very easily. Oh, sorry, I was just going to go over a quick memory tool that I use for doxycycling. So doxycycling kind of sounds like cycling. And, you know, when you're riding on your bike, you can fall over and break your bones. And that kind of relates to the effects on depleting calcium and on bone growth. So it's a bit of a stretch, but, you know, might work for you. The next antibiotic is gentamicin. So this is an aminoglycoside antibiotic inhibiting protein synthesis by irreversibly binding to 30S. So as opposed to doxycycline, which is bacteriostatic, gentamicin is bactericidal. So it's a bit more intense and indicated for a bit more severe infections. So gram-negative bacteria or aerobes more specifically. And it has more severe adverse effects as well. So nephrotoxic, so toxic to the kidneys and to the ears as well. And another memory tool that's been going around is if you think of a very sad man who has kind of sick or sore kidneys and a sore ear. So the man refers to gentamicin. The sad part is, you know, he's a bit negative, gram-negative bacteria. Yeah, it's a bit of a stretch, but whatever. And then he has sore kidneys and a sore ear, so it relates to the adverse effects. So sounds a bit strange, but sometimes it's the strange ones that kind of stick in your mind. And the next ones for protein synthesis are erythromycin and clarithromycin. So these are macrolides. And they reversibly bind to 50S, so the larger ribosomal subunit. Again, they're bacteriostatic rather than cidal, and they do have cardiac side effects, so they can cause arrhythmias. So really important not to prescribe these for patients who already have existing heart conditions. So not, I guess, as relevant now, but just, you know, to keep in the back of your mind for the future. And the next one is chloramphenicol. So honestly, I kind of learnt about this one from revision lectures last year. I never really heard it from any lectures. The main buzzword that comes up in relation to chloramphenicol is grey baby syndrome. So this is an antibiotic. It does inhibit protein synthesis, not as important as the other ones, but because the antibiotic can't really be processed properly in the liver of babies, it can, you know, cause them to turn grey or have a grey appearance. So if you hear grey baby syndrome, just think of chloramphenicol. Or I guess use a process of elimination from the other drugs that you might know a bit better. And so for that reason, you wouldn't really give it to children under 2 years or pregnant women. So which of these antibiotics will inhibit nucleic acid synthesis? So I'll just give a moment for that. You can type in the chat. I'm hearing some mixed responses. Okay, so we have A, ciprofloxacin, metronidazole, and also rifampicin as another one of those ripe drugs. Actually, I don't think rifampicin should have been there, but you know, we'll move on. Actually, no, that's fine. Okay. So we have metronidazole, which is a nitroimidazole antibiotic. So same ending as well, makes it a bit easier to remember. So in terms of inhibiting nucleic acid synthesis, you don't really need to know too much information about how exactly it inhibits nucleic acid synthesis. The ePharmacology website really goes into this, but I've seen a couple of questions in the chat kind of asking how much detail we actually need to know about this, and knowing what it inhibits and not exactly why is perfectly fine and probably more than enough. So just the fact that it inhibits nucleic acid synthesis. And this is used in anaerobes and also can be used for parasites such as Giardia. So anti-protozoal effects as well. Important kind of buzzword adverse effect is the metallic taste. So you might get a metallic taste in your mouth after you've had metronidazole. And the way I remember it is just metronidazole has, and then metallic taste. So the same first 3 letters. The next one is ciprofloxacin. So, sorry, it's a bit of a typo. I should say nucleic acid synthesis, not protein synthesis. But it's a fluoroquinolone, and it stops the topoisomerase enzyme. Doesn't really come up too much. Yeah. So I'll just try and, I guess, keep on top of time. I know there are a lot of antibiotics. Okay, so the next one is rifampicin. Again, this is part of the ripe drugs. So it is an RNA polymerase inhibitor, so preventing DNA transcription and translation for people who have tuberculosis infections. And then a buzzword adverse effect is the red or orange secretion. So, you know, urine, saliva, all that kind of stuff. It's a pretty distinct, memorable adverse effect to remember for rifampicin. So R for rifampicin, R for red or orange secretions. And yeah, this is just going over the ripe drugs. So rifampicin and isoniazid, definitely the more important ones, but you can also have pyrazinamide and ethambutol as well. So why might multiple drugs be needed to treat tuberculosis? Yeah, a few more answers in the chat. Yep, so we want to really make sure that we're eliminating the TB completely from the person's system, and also therefore eliminate resistant strains from arising. So good job to everyone who got that, which is good. However, the ripe drugs in general are hepatotoxic, as I mentioned, and then rifampicin has that unique adverse effect of red-orange secretions. Okay, so the last class of antibiotics is the folic acid or the folate synthesis inhibitors. So which ones would they be? Yep, so we have methotrexate, trimethoprim, and sulfones or sulfonamides. So importantly to remember as a general rule, rather than remembering it one by one, that everything that disrupts folate synthesis is just bacteriostatic. Okay, so we have sulfones or sulfonamides. Honestly, haven't really seen this too much since learning about it in first year, but this inhibits the para-aminobenzoic acid from being converted to folate and is bacteriostatic. Again, probably don't need to know this level of detail, but you know, just for your information. The next one is methotrexate and trimethoprim, and these have pretty much the same mechanism action, and by far the one you would hear about the most is methotrexate. So this is a dihydrofolate reductase competitive inhibitor. So it's inhibiting the enzyme that converts folate to the purine nucleotide, so guanine and alanine. And as you probably remember from studying the cancer pharmacology, methotrexate is also used not only as an anti-microbial, antibacterial, but also in anti-cancer because it is able to suppress the immune system and has anti-inflammatory, I guess, functions as well, which is pretty cool. Okay, so these are just, I guess, a summary of some buzzwords that often come up, and it's a good thing to kind of go over as you get closer to the exam. So I've mentioned all of these before, so I guess you can go over that in your own time if you'd like. Okay, so well done everyone for getting through antibiotics. I know it's a lot, it can be a bit overwhelming, but now this is kind of a bit more chill, just antivirals. There aren't too many of these. Some important considerations though is that these are not broad spectrum like antibiotics. The antivirals that we have are only able to target the one specific infection that they were designed for. And there are also fewer drug targets. And that's because, as we know, viruses, they hijack our own cells and use our own organelles against us. So we don't want to implicate any of our own organelles in drug targets. So all antivirals are actually virustatic. So they only stop the growth and then rely on the host immune system to actually kill the viruses. And resistance resistance to antivirals also could be a really big problem in the future, especially the viruses, the RNA viruses, because they have no mechanism of proofreading or repairing mutations that occur in replication. So that's why they're not really used too much. Okay, so I'll start off with, I guess, the biggest topic, which would be HIV antiretroviral therapy, so ART. And so there are a lot of different classes of HIV antivirals. Yeah, you definitely don't need to know too much detail, all these, apart from just recognizing that I guess they're part of antiretroviral therapy. So we have nucleotide reverse transcriptase inhibitors, non-nucleotide reverse transcriptase inhibitors, protease inhibitors. So this protease inhibitors inhibit the protease enzyme, which cleaves parts of the virus to make them functional. So often proteins need to be cleaved to become functional. So this stops that process. And we also have INSTIs, so integrase strand transferase inhibitors. So this inhibits the integration of the HIV into the host genome. We also have fusion inhibitors and entry inhibitors. In terms of general principles, I think this is quite important, is that the antiretroviral therapy for HIV is not curative. The goals of therapy are to reduce transmission, to maintain and restore the host's immune system and immune function, to reduce the viral load as much as possible. A lot of the drugs have really severe multisystemic side effects, so it's definitely not an easy thing to go through. And I guess to control the viral load as best as possible, and also I guess to prevent resistance as well, is combination therapy is often used. So at least 3 drugs from at least 2 classes is a good thing to remember. We have 2 types of prophylaxis. We have pre-exposure PrEP and post-exposure. So an important pre-exposure drug is Truvada. So that's a combination between 2 antiretrovirals, tenofovir and emtricitabine. Both are the nucleotide reverse transcriptase inhibitors. So that's just, basically inhibiting the viral reverse transcriptase. Okay, so ritonavir is the main HIV drug that we kind of learnt about, and it is a protease inhibitor, which I described just before on the last slide. An important mechanism of action, apart from its activity as a protease inhibitor, is a pharmacoenhancer. So what I mean by that, it actually slows the metabolism of other HIV drugs, which is really important, because if we're slowing the metabolism and the rate of deactivation of other HIV drugs, we don't need to prescribe as high of a dose. So we can prescribe a bit of a lower dose, which means there's less chance of resistance and also fewer side effects, which is really good. However, it does have hepatotoxic and kind of cardiotoxic adverse effects. A memory tool that was going around for this one is ritonavir. So Rito kind of sounds like Doritos. There's no protein in Doritos. It's a protease inhibitor. So yeah, again, bit of a stretch, but whatever. The next one, honestly, pretty low yield. The main point of zidofudine, or it's got another name that's abbreviated to AZT as well. It stops vertical transmission of HIV, so from mother to child. Yeah, not as high yield. Okay, so this is another antiviral, oseltamivir. So which viral infection is this used for? Feel free to drop in the chat. Yep, keep it coming. Cool, great. So influenza A and B. Don't worry too much about whether it's A or A and B, like A and B are just kind of the general ones anyway, so influenza is fine. How does oseltamivir fight influenza infection? What does it actually do? Yep, seeing a couple good answers. Great. Okay, so it's a neuraminidase inhibitor. So it prevents the release of the replicated virions from the host cells. So we have two main antigens on influenza, the hemagglutinin, used for viral entry, and the neuraminidase, which is used for the exit. So yeah, don't mix those ones up. Okay, so this is the diagram, which is kind of, if you're more of a visual learner, can help you remember the action of neuraminidase and oseltamivir. So as we can see, it just cleaves the receptor that's kind of holding the virion to the host cell and then allows its release. So preventing the virus from getting released. Okay, so next we have acyclovir. So which viral infection is this used for? Or I should say, which class of viruses? Yeah, good job, everyone. Herpes viridae, so herpes simplex 1 and 2, and also varicella zoster, so chickenpox, which is also part of the herpes viridae family of DNA viruses. But yeah, more so the HSV. And so what is its mechanism of action? How does it actually fight an HSV or VZV infection? Yep, it's okay if you don't know. Yep, it's a guanine analog, so it's going to prevent DNA synthesis. So it's acting as, yep, kind of a fake guanine nucleotide and stopping the DNA from being replicated. And the last antiviral is interferon alpha. So this is a biological drug. It's a cytokine that naturally exists in our own bodies, but we're able to administer, so kind of synthesize that, artificially and administer that to patients. So it's going to, because it's a cytokine, it's going to exacerbate our own immune response. So to enhance, repair, phagocytosis, or the migration of leukocytes to the, affected site. And it also has a role in interfering with viral mRNA. So it's pretty useful. And because it's a biological drug, it's kind of not foreign to our body, which is quite nice. Okay, so the next class is antifungal drugs. I'm just going to go over the unique structures that these drugs target, just so it makes a bit more sense. So the unique structures in fungal cells that we don't have in our own cells, the first one is ergosterol. So this is the alternative to cholesterol that's present in our cell membranes. So funguses have ergosterol in their cell membranes to maintain that stability and fluidity and all that stuff. And then in cell walls, instead of the proteoglycans in bacteria, funguses have chitin, mannose and beta-glucans. So these can all be specific drug targets because they are unique to funguses. Okay, so the first class of antifungals, so this is, just the broad spectrum, regular old antifungals. So this is fluconazole, so an azole antifungal. As I just mentioned before, it's going to target the ergosterol, so the cholesterol equivalent in the fungal cell wall, which we can see here on the diagram in the right. So therefore, it's going to disrupt the fungal cell membrane. It can be used as a topical cream or even orally as well. It's just used for, tinea thrush, fungal nail, day-to-day fungal infections. It's pretty well tolerated, not too intense. Yeah. The next two antifungals are for more severe systemic fungal infections, which I guess is an important distinction to remember. This is amphotericin B, which is a polyene antifungal. Again, it's going to bind to the ergosterol in the cell membrane, but it's going to form pores, and that's going to result in lysis and the death of fungal cells. So it's a bit more intense. And as I said, it's going to be used for more systemic fungal infections like aspergillosis or candidiasis. And because it has a more intense mechanism of action, it does have more intense side effects as well. So nephrotoxic, it can also result in fever and chills. And the last antifungal drug is caspofungin, and so it's part of the echinocandin class. It's going to inhibit glucan synthesis. So if we see on this diagram on the right again, we can see the glucans here are part of that fungal cell wall. So it's going to disrupt the cell wall, again indicated in the more severe systemic fungal infections and with similar adverse effects as well. Okay, so I think the last section I've got here is anti-cancer. Okay, so we have two main classes. We have the classic anti-cancer and then the more novel anti-cancer. And this can be divided into non-specific. For the classic anti-cancer drugs, there are lots of side effects. They just target all rapidly dividing cells, whereas the novel anti-cancer drugs are more specific in action and are often associated with fewer side effects, which is really good. Okay, so the first core drug in the anti-cancer category is the alkylating agent. So cyclophosphamide is an example of that. It's going to form covalent crosslinks between the guanine nucleotides. So that causes the DNA strand to break and then apoptosis of the cell. As I said, it's non-specific. So it results in many systemic side effects because it's going to target all rapidly dividing cells. So it's kind of the typical, I guess, cancer side effect profile of hair loss, infertility, kind of suppressed immune system, that kind of thing. And this is the same for all of the classic anti-cancer drugs. A memory tool that I use to remember this is cyclophosphamide. So the guanines, they want to be close, they're a family, they're going to form cross links. But yeah, that's kind of its mechanism of action. Cool. The next class of classic anti-cancer drugs is the cytotoxic antibiotics. So it's pretty much in the name. So it's just a really, really strong antibiotic it's too strong to be justified in normal bacterial infections. So it's kind of saved for the more intense cancer therapy. And so an example of that is doxorubicin. It inhibits the transcription and translation of proteins. And something important to consider in the classic anti-cancer drugs is which stage of the cell cycle is this drug going to be effective in. And so because it's inhibiting the transcription and translation of proteins associated with the growth and replication of all the organelles, it's going to be effective in the G1 and G2 growth stages of replication. In contrast, we have methotrexate, which is the antibiotic that we mentioned before, and 5-fluorouracil, which are antimetabolites. So these prevent the DNA synthesis or the replication stage. So methotrexate, as we mentioned before, is a dihydrofolate reductase inhibitor, preventing the nucleotide synthesis. And 5-fluorouracil is a direct nucleotide analog. As we know, uracil is a nucleotide, so 5-fluorouracil is kind of an adapted nucleotide, which will block the normal DNA synthesis. And because it's affecting the replication of genetic material, it's going to be active in the S stage. Okay, so then we have anastrozole and tamoxifen. So anastrozole is an aromatase inhibitor, so it kind of starts with the same letter. And aromatase enzyme usually converts androgens to estrogens. So don't worry about this too much. I know you probably haven't gone into it much at all, so that's perfectly fine. But basically what it does, it inhibits estrogen. And so it's then indicated in estrogen-dependent breast cancer. So some types of breast cancer rely on estrogen to stimulate further growth and replication. So if we're inhibiting estrogen formation, it's going to help stop the growth, which is really good. And memory tool, anastrozole, Annie, again, yeah, a bit of a stretch, but helps with.
Can we actually?
Sorry, did someone have a question? Or I can't see the screen, but maybe if you had a question, just type in the chat because I didn't quite get that. Just looking at the chat now, I did see a question, do we need to know the side effects for these anti-cancer drugs? So as I mentioned on the initial slide, that all of the non-specific ones have this similar side effect profile of hair loss, immune suppression, potential infertility, that kind of thing. But no, not too much detail, I don't think. And the second anti-cancer drug that's useful in estrogen-dependent breast cancer, again, is tamoxifen. So this is a SERM, so a selective estrogen receptor modulator. So where anastrozole actually stops estrogen from forming, tamoxifen stops estrogen from being able to bind to the receptors on breast tissue, again, reducing the proliferation and the growth. And so and memory tool is TAMI, so it kind of sounds like girls' names which are used in breast cancer. So even though we know that breast cancer can also happen in males as well. Okay, so this is the main core drug for the novel anti-cancer category. I know you might have come across more as well, but this is the only one that's listed on the ePharmacology core drug list. So just because of time, I'm not going to go over all the ones that you might have learned otherwise as well. So why do novel anti-cancer drugs have fewer side effects than the classic drugs? So I'll be taking answers in the chat. Oh, I already released the answer, that's fine. So novel anti-cancer drugs, most of them, or pretty much all of them, are monoclonal antibodies. So rather than targeting all rapidly dividing cells, they target specific receptors on structures only present on cancer cells and not healthy cells. So because they're not attacking healthy cells or rapidly dividing cells, we're not going to have the same systemic side effects, which is really good. Okay, so the drug we're going to talk about here is trastuzumab. I definitely can't pronounce that, but it targets the HER2 receptor. So that stands for the human epidermal growth factor receptor 2. So it's indicated in HER2 positive breast cancer, and the kind of explanation behind that, which is mainly so you can actually understand it hopefully, but don't stress about memorizing it too much, is that this receptor, the human epidermal growth factor 2, is overexpressed in about 20% of invasive breast cancer. So because it's a growth factor, we want to block that. We don't want to be promoting the growth. So if this monoclonal antibody is blocking this growth factor receptor, we're going to reduce the proliferation and increase survival, hopefully. However, does have cardiac adverse effects. Again, not a major point. As a memory tool, the drug trastuzumab kind of sounds like tutu, and it binds to the HER2 receptor. Cool. Okay, so I'll just do a quick time check. Yeah, so I've gone a bit over time, so maybe just practice these questions in your own time, but I really hope that made sense and clarified things for you. And yeah, these slides will be on the Google Drive, hopefully. So yeah, thank you for coming and for your participation in the chat as well. I'll answer any questions. Yeah.
Julia, if you want as well, we can probably, I guess, like take 10 minutes and go through the questions, if that's okay with that.
Oh yeah, sure.
Yeah, so we might as well just, go through the questions.
Yeah, I only had like a couple, so might as well. Thanks for that. I know it's so pushed for time. So these just relate to antibiotics because I guess they're the most amount of practice questions for antibiotics, and it can be a bit of an overwhelming topic as well. So again, I'll just be taking answers in the chat, keep them coming. So 16-year-old Mark is about to undergo surgery. So which medication would you give to relax his muscles? I'll give it a bit longer this time so that more people can reply. If you don't know, that's fine as well. It's like a bit of information overload. Cool, rocuronium. So yeah, this is our non-depolarizing blocking drug. So really good for muscle relaxation. So a couple people said pyridostigmine. So that's going to actually increase the concentration of acetylcholine at the neuromuscular junction. So that's going to lead to increased muscle contractions rather than relaxation. Okay, So next question, Christina is a 55-year-old Southeast Asian refugee with a lung infection. You prescribe her an antibiotic, advising her not to be alarmed if she experiences orange urine, as it's just a side effect of the medication. Which drug must you have prescribed her? Yeah, everyone's really on top of this. Good, cool. Rifampicin, so one of the RAP drugs. Okay, so Megan is traveling to Cambodia to find herself during the uni holidays. Which drug would you prescribe her as malaria prophylaxis? Again, I'll give it a bit longer. Everyone feel free to jump in. Yep, G-doxycycline. So this has really multiple indications outside of just being antibiotics. So acne and malaria is two such examples. Okay, so what are Emma and Megan at risk of now that you've prescribed them doxycycline? Cool, all of them. So I know I haven't mentioned GI upset in the actual slides. That's pretty much just a side effect of everything. So yeah, when in doubt, say GI. Yeah. Sam was treated for MRSA in hospital, but to the doctor's surprise, he turned quite red. What was he treated with? Okay, good job, everyone. Vancomycin. Okay, thank you again for listening. I'll go back through the chat because I know some questions were asked along the way that I would have missed. So I'll reply to you guys individually. But yeah, thanks.
Thank you, Julia. That was a massive effort, a lot of stuff there to go through. So thank you so much for giving the year ones that. But next up is Yajat, who's going to take us for Microbio+Immunology. So we'll hand it over.
Hey, thanks, Bhuvi. All right, share. Wait, can you guys? Yeah, okay. There we go. All right, microbiology and immunology. Once again, a disclaimer that these slides are all remixed from all those sources there. Pretty much our PSPs that we had last year, whose slides are still available for you guys, I believe, if you guys want some extra stuff, and our revision lectures from last year. All right, this is an overview of what we're gonna do. We'll start with all the pathogens, bacteria, viruses, parasites, fungi, methods of control, innate, and then onto immunology, innate and adaptive, and then some practice questions at the end. All right, big picture stuff, prokaryotes versus eukaryotes. This is just a review, so pretty much prokaryotes are smaller, they have a single cell, these shapes, whereas eukaryotes are bigger, can be multicellular, but they can also be unicellular. So unicellular eukaryote is a protozoa. No nuclei in prokaryotes, no membrane-bound organelles. And then eukaryotes don't have a cell wall, while prokaryotes do. And then these are just examples. And then for reproduction, prokaryotes are only asexual reproduction, which is binary fission, and eukaryotes are either or. So over here I mentioned there's three main shapes for prokaryotes. So these are the three. You've got your cocci, which could be like your Staph aureus in great like clusters. You've got your bacilli, which is your like E. coli, salmonella, and then you've got your spiral shaped. This is just a diagram of how binary fission occurs, so asexual reproduction in prokaryotes. Yeah. All right. Regarding host-parasite relationships, there's these three. So you've got mutualism, where between the bacteria and the host, so the host we usually talk about are humans, there's mutual benefits. So this is like your normal flora in your digestive system, on your skin and stuff, which we'll go through more. You go commensalism, which is one, like either the bacteria or the host benefits, and then there's no harm or benefit really close to the other. So it's one-sided. And then parasitism is one benefit at the expense of another. Yeah, so normal flora are either the first two, but once they do invade, so like your skin, bacteria on your skin entering your blood, or in like immunocompromised or diabetic patients, when normal flora becomes harmful, it can become parasitic and then cause infections. All right, starting off with bacteria. The highest yield things in bacteria is regarding their cell walls. So when you do gram stain under a microscope, there's either gram positive or gram negative, which depends on how the thickness of the peptidoglycan layer. So in gram positives, there's a very thick peptidoglycan layer. In gram negatives, there's a thin one. And so gram positives turn up purple and gram negatives turn up pink. I think one of our PSPs last year told us that like they remember that because in like stocks, like when stocks are going negative, they go red. And so like gram negative is like pink, which is more of red. Whatever helps. So here are some examples: Staph aureus, strep, your two big name bacteria will be purple, gram positives. Cholera, Vibrio cholera would be more pink, gram negatives. And then there's gram zero or mycobacterial bacteria, which don't show up on gram stains. And because they have this waxy lipid layer of mycolic acids, and so we use acid-fast stains. And the biggest example of this is Mycobacterium tuberculosis, which causes TB. And it goes red with acid-fast stains. All right. Structures of bacteria. This is just a table doing a big overview. There'll be a lot of tables in this presentation because that's just how I like to learn. I would like take this and then put it into Anki and then put like an image occlusion and try to recall them if it just works if any of you are into Anki. All right, flagella it's like pretty much for movement. You can have single or multiple flagella. It's those little whip-like structures at the end of the bacteria made of flagellin proteins. Capsules help for avoiding immune detection. So it's one of the virulence factors for bacteria. And it also helps for like adhesion. We'll go over what help bacteria survive. But yeah, it's a sticky, sugary layer around the cell wall. Pili, fimbriae, adhesion molecules. These are like little hair-like structures coming out of the cell surface, and it helps with adherence, and it also helps with biofilm formation, which is like if you have a catheter or in general a surface, they kind of talk about it more so with surfaces that are man-made, that are in humans. It's like a hip replacement surgery or like a catheter, because it's such a smooth surface that you don't really find often in the body. A lot of bacteria can form these biofilms, which is just a thin layer of a lot of bacteria, and they set up that colony and they can reproduce really fast. And so adhesion molecules help with that. Spores, some bacteria make them, and like tetanus, and they have some information, and it helps with spread as well, I'm pretty sure. Yeah, so virulence factors, just adding on to that, you can classify them in 3 categories. Either they stick, damage, or evade immune destruction. So sticking to surfaces is adhesions, fimbriae. Damage is by exotoxins, where bacteria will release toxins, and that'll cause like an immune-mediated damage to the host. And with the capsule, it can evade immune destruction because, like, if you remember, it's just like a sugar layer on the outside of the bacteria, and so our immune cells don't really recognize it as much. All right, this is a really important flow. It's pretty intuitive, but basically this is how bacteria cause damage. So first they enter the host via transmission, enter into the body. So they have to get past our innate immune defenses, like the skin. They will have to overcome our own immunity and then find somewhere to grow and spread, and then eventually they can cause damage. And so we want to break the cycle somewhere if we want to prevent infection. So the most effective is like preventing entry into the body or transmission. Opportunistic infections. Okay, so we have a normal flora with tons of bacteria, commensal bacteria that's useful for us. But in these kinds of cases of chemotherapy, HIV, AIDS, which is immunosuppressive, pregnancy, and these kinds of factors, the normal flora can actually turn harmful. So some examples are E. coli, which is normal flora in our GIT, Staph aureus, which can cause skin infections, and Candida, which is a fungus, but it can cause upper respiratory or genital infection if the normal flora is wiped out. All right, I know in your lectures, Meredith kind of just goes with a bunch of bacteria and it can be overwhelming. These are the main bacteria that I would suggest knowing. So again, like this table, if you use Anki, you can just image occlude all these things and then try and recall it. But yeah, we talked about mycobacterium. And then the gram-positive, gram-negative can be useful. Over time, if you see like enough pictures, you kind of start like visually learning that like, okay, E. coli, and if you see like slides of E. coli, they're pink, gram-negative rods. If you see like Staph aureus, it's a very typical, very unique structure of like purple clusters of cocci. But yeah, so just go through these and get to know it. Microbiology can be quite a bit of memorization. These are just organized by like different areas and different causes and where they can be acquired. So we've got like your tetanus, Clostridium tetani and bites, genitourinary tract causing your UTIs is your E. coli, lots of gram-negative bacteria. You got lots of Staph aureus and strep like on your skin. So anytime there's a wound, it can enter the bloodstream. Clostridium perfringens is for gas gangrene. It's an anaerobic bacteria. There's some more. And like over the years, like you'll go through these a lot more as you go into system-based learning. So like in respiratory, you'll go very in-depth in all of these. So just have an overview understanding like pneumonia, strep pneumonia, like everyone knows pneumonia is lungs infection, so it's more respiratory. All right. Check the chat. Yeah. Okay. Yeah, we've pretty much gone through this. Normal flora is mostly commensal, some mutualistic. Shortly, yeah, so when it's acquired, and then this is the most important. So provides protective, structural, and metabolic functions to the host. So like the most useful example people talk about is the gut microbiome. And when that microbiome gets replenished or gets like erased because of antibiotics or stuff, it can cause some gut problems. Okay. So skin, mostly gram-positive. I've seen some exam questions on this, just know it's mostly gram-positive. GIT, they have to be acid tolerant because there's a lot of acid in your stomach. And the bacteria in your intestines increase as you go from the stomach down. The pH increases because there's less and less acid, and so more bacteria can survive there. Genitourinary tract bacteria from skin, colon, and vagina can enter into the urinary tract. Upper respiratory is more of a mix, and these 1, 2, 3, 4 places in the body are important to know because they're completely sterile. So in normal situations, there should not be any bacteria. So that's your lower respiratory tract, which is your like alveoli into your lungs and stuff. Your upper urinary tract, which is around your kidneys. Your central nervous system, brain and spinal cord and stuff, and then your blood. So these under normal circumstances should not have any pathogens. All right, moving on to viruses. So viruses are non-cellular and they're obligate intracellular parasites. So they need to enter into a host cell in order to replicate and survive. The general structure is nucleic acid sort of single-stranded RNA, double-stranded RNA, DNA, either or, surrounded by this outer protein coat, which is a capsid, and that can be of different shapes and is used to like classify viruses. And then it's surrounded with, it may have a lipid envelope, which it could get from host cells. And then it could also have this glycoprotein layer on top. Is that all? Yeah, okay. Yep, so as we talk about classification is based on the nucleic acid type, the shape of the capsid, whether or not it's enveloped in a lipid envelope, and then which like hosts it can invade. So like classic examples, influenza. So influenza A can invade like humans and animals, which makes it more prone to mutating, and then which disease it produces. Retroviruses are always single-stranded RNA viruses, and so they use this enzyme reverse transcriptase to essentially integrate their genetic information in host DNA. So normally we transcribe the DNA, but this does it the other way. So it usually goes DNA to RNA, but with reverse transcriptase, these viruses can put their RNA back into DNA, and it forms a cDNA, which can enter the nucleus and then fuse with the host cell DNA, and it kind of just makes this virus-producing factory. Okay, important viruses. We got RNA and DNA. This is an overview. So flu, as we talked about. So type A can cause pandemics because it can infect humans and animals. So there's more hosts allowing it to mutate more. Antigenic drift, antigenic shift. So drift is like small changes, like something's drifting. I kind of think of like icebergs drifting. It's really slow, and so it's small changes that accumulate over time. Types A and B do that, and then antigenic shift is like a big change in the virus, and that results in a whole new. It's mainly hemagglutinin, which we'll talk about later as well, which is allows entry of the influenza virus, and this is type A. But this is what can cause pandemics. That's why you see like the H1N1 and all the different flus that go across the world. I don't think COVID's part of them. Measles, rotavirus, mumps are RNA, and then DNA, you've got your hepatitis B, herpes, and human papillomavirus, which can cause warts. And again, there's another table. So yeah, this kind of just is your main viruses that you want to know, and then what they cause. Yep. Okay. Well, replication. Okay, so this is pretty much the life cycle of how viruses enter, cause damage, and release. So they attach usually by a receptor on the host cell, and then they are absorbed into the cell, so entry, and then they hijack a host cell process. So they, like we talked about reverse transcriptase, there's a bunch of different mechanisms that viruses can do once they're in the host cell that they can replicate. They assemble themselves by forming capsids around the nucleic acid, and then they release. And during the release, you can see over here, like in the exocytosis, there's like a part of the human cell, the host cell membrane coming along with it, which is forming the lipid envelope. And so as you can imagine, this would be the host's own cell membrane. And so I'm pretty sure it can help with like evading the immune system as well. All right, moving on. Fungi. They have to be aerobes. They're everywhere, but they're not always pathogenic. Fungi can undergo sexual and asexual reproduction. And the key characteristics, so they have a cell wall as well, made of all these things. The most important one that they highlight is chitin. And this is very buzzwordy, is a cytoplasmic membrane made from ergosterol, while humans have cholesterol. And so this is a mechanism of action of many antifungal agents. So you have your three classes. You have your yeasts, molds, and dimorphic. And these are some examples. So dimorphic exists in both, so like molds thrive at 25 degrees and yeast thrive at 37 degrees Celsius. There's the mold or cold, it rhymes, so it works. And then allergies. So in mold fungi, they have hyphae, which can release spores and disseminate into the environment. And so if you inhale spores or cells from the mycelium, that can cause allergies. All right, these are your big name fungi. So you've got three major groups: cutaneous, subcutaneous, and opportunistic. Your cutaneous dermatophytes need keratin, and they're your ringworms, your tinea pedis, curris, corporus, or athlete's foot, jock's itch, and stuff. Subcutaneous need to invade under the skin, so it's inoculation. These are more serious, and the sporotrichosis from rose bushes is a big one at Monash for testing. And then opportunistic are pretty much, they're very common, but they only really present in immunocompromised patients or patients in long-term antibiotic use. So your candidiasis, which is a yeast thrush-like infection, aspergillosis, which is allergy, and it's usually in your respiratory system, and then pneumonia. Fungal pneumonia is very prevalent in AIDS or cancer patients. And again, tables. Yeah, you can go through this. All right, parasites. Parasites, yeah, so they're eukaryotic. There's three broad categories, and protozoans, helminths, and ectoparasites. So protozoans, we talked about them earlier, they're single-celled eukaryotes. Helminths are worms, and ectoparasites live on skin. All right, this is a one-slide summary of all the parasites you need to know. All right, helminths, you got hookworms, which live in your small bowel. They cause anemia. Enterobius, they're in the cecum, so that's part of your large intestine, and they crawl out of the anus at night to lay eggs. So there's like, I'm pretty sure a sellotape test that it's very common in children, and so like in the middle of the night, if you use sellotape, you'd see like little white dots on that. Filariasis, it lives in lymphatics and it causes elephantiasis, which I'm pretty sure is just a lot of edema and it causes swelling. Trypanosoma is your African sleeping sickness. And then your three very high yield ones. One is malaria, so it's Plasmodium. And then you've got these 5 species, which you might as well know, but the most important was Plasmodium falciparum. Malaria infects the blood and CNS, and we talked about those two being sterile fields, so we want no pathogens, and that's what malaria infects. Prevention is key, so prophylaxis, nets, mosquito repellent, mosquitoes are its vector. So no fleas mosquito is how malaria is transmitted. Giardia, very buzzwordy, are greasy stools. So you inject these little cysts in water and it can cause bloating, nausea, abdominal pain, treated with metronidazole. But yeah, anytime there's greasy stools, like you'll keep seeing these questions, it's always Giardia. And then Toxoplasma gondii is cat litter, kitty shit. But there'll be like Monash questions where it's like someone's found unconscious and there's cats around them, and what is the most likely pathogen, and it'll be Toxoplasma gondii. All right, control of microbes. All right, we'll start with antibiotic resistance. So going back to bacteria, there's 5 major targets that our antibiotics are used. So that you would have probably covered that in Pharm: cell wall, protein, nucleic acid, metabolic pathways, and cell membrane. And this is the MIND-ME acronym. It's really worth knowing it in preventing resistance. So microbiology guides therapy whenever possible. It's pretty intuitive. Indications should be evidence-based. Again, makes sense. So you take the narrowest spectrum required. So basically you're not taking antibiotics longer than you need to. Dosage is appropriate. You want to minimize duration of therapy. Oh yeah, narrow spectrum, sorry, it's not about duration. It's about like broad spectrum versus specific drugs. So like your amoxicillin and stuff are very broad spectrum, and so they'll wipe out a lot of bacteria as opposed to getting very specific antibiotics for the specific bacteria. But obviously that takes time. because you need to do culture and stuff to figure out what bacteria is causing the disease, and ensure monotherapy in most cases. So just know these. They'll test you like with opposites and be like, which one is true? And they'll be like, use the broadest spectrum possible, or use as many drugs as possible. And then you just got to know that those are wrong. All right. So there's these three main mechanisms of how antibiotics develop resistance. There's innate. So some bacteria just genetically can survive antibiotics. And these are ways that they can. Lots of lipids or waxy membrane. So Mycobacterium tuberculosis, again, it has that really waxy lipid membrane. And so normal like antibiotics don't work against it, and you need your ripe. Acquired resistance is these four ways. So basically changing the places that antibiotics work for so that they don't work anymore. And non-genetic is like literal physical ways to survive. And so like in an abscess, you can wall off, like bacteria can wall themselves off so the immune cells there, you can't access them. Or if they can survive without a cell wall, because a cell wall is a huge target for a lot of drugs, or biofilm formation. Just check the chat. Okay. These are ways resistance can be acquired in bacteria. So transformation, conjugation, transduction are the big three. Transformation is taking DNA from the environment, because DNA pretty much determines everything about how bacteria acts and reproduces and stuff. And so that's what's going to be the primary way of getting resistance. So if you take DNA from the external environment, it's transformation. Transferring DNA between two bacterial cells is conjugation, and a virus infecting a bacteria is transduction, and that's called a bacteriophage. Plasmids are these little donut-shaped DNA molecules. They're double-stranded, and they're separate from the normal bacterial DNA. And so these are what are used to transfer through conjugation. And they also code for their own replication, like in their DNA. So it's self-replicating. It doesn't need the original DNA to replicate. On beta-lactamase, I don't know if you talk about in pharmacology, but it's plasmids are in charge of making beta-lactamase, which can render like penicillins and amoxicillins. They make them not work because they break the beta-lactam ring. So those plasmids are in charge of making those antimicrobials. And transposons. They are these little genes. They're called jumping genes, and they kind of encode their own movement. So they can move either plasmid to plasmid, plasmid to chromosome, or chromosome to chromosome. And there's these two types. So there's insertion sequences, which are just a few nucleic acids or a few base pairs. They're very small, but the ones we're mainly concerned with are composite transposons. And they're the ones that carry the antibiotic resistance genes, the virulence factor genes, and so they can cause a lot of damage. Agar plates, I remember being very confused by this in the lectures, but they're really the highest yield one is horse blood agar, and just know these three, they can show up. They detect hemolytic activity. So some bacteria have the ability to lyse red blood cells, which can cause a lot of damage, as you can imagine, in humans. So a horse blood agar kind of is useful in determining that. So beta hemolysis here in the middle is complete hemolytic, like complete lysis, complete digestion of red blood cell contents. And so you'd start with this red, and then you'd have this lightened clear zones. So an example of this is strep pyogenes. We learned about like hemolytic strep pyogenes and stuff. Alpha hemolysis is partial lysis. So you have these green, brown zones from your strep viridans. And gamma hemolysis is when a bacteria doesn't have any hemolytic activity, so it stays the same. Okay, this is how infection happens. This is the big chain that happens. So you start with your infectious agent, the four main groups we talked about. they have a reservoir specific. So that's where the agent survives and replicates. So it can be humans in most of them that we talk about. Some can go into animals like influenza A. Vibrio cholera, for example, its main reservoir is like in water, and it can be spread through that. It can like survive in water, so it can go into food and stuff. Now, portals of exit from the reservoir, normally it'll be like how it's excreted from humans, but it can be through other ways. So respiratory, think droplets, secretions, GIT infections, think your fecal oral route. And then that's going on to your means of transmission. So droplets, contact, etc. How it enters the host, either through skin, mucous membranes, oral route, and then your susceptible host. So there can be tons of bacteria that can get all the way into the host, but if the patient has a strong immune function, it won't cause any problems. And so then you really want to worry about your immunosuppressed, your cancer patients, pretty much patients with any pre-existing conditions, like patients with AIDS as well, who are immunocompromised. Okay, so you're in controlling infectious agent. This is pretty important in hospitals. There's these three, it's basically a spectrum where you start with cleaning, disinfecting, and lastly sterilizing. Cleaning is pretty much just hot water and soap by washing, soaking. Disinfecting, we'll go on to like all the different ways you can do that. But disinfecting is killing most viable organisms, so it's not a sterile field, and that's like your boiling or chemicals. And then sterilization is pretty much anything that penetrates your epitheliums, any scalpel, any drip, any cannulas, they have to be sterilized. That's 100% of viable organisms and spores will be killed off. These are your three main ways for disinfecting. So moist heat, its main thing is inactivating proteins, and it can't really kill spores unless it's over 100 degrees. Chemicals, you've got these 5 chemicals that you want to know, and UV radiation. So these all are disinfecting. So they're not getting rid of they're getting rid of most of the pathogens. And sterilizing, again, we have moist heat again, we have some chemicals again. But yeah, dry heat, just know these temperatures and time. So 170 for 1 hour, 160 for 2 hours in dry heat, and then moist heat and pressure is a lot faster. So it's only 3.5 minutes or 15 minutes at lower temperatures. So the most effective out of these methods, it really depends on the most effective out of these 5. Yeah, so for sterilization, sorry, that's just a question in the chat. It depends on what you're doing. So some substances, like some medical supplies, can't go into like these really hot circumstances. Like think of gloves and stuff. They're very heat sensitive, so you want to do ionizing radiation. So it's really a case-to-case basis. But there are some examples of stuff that are used. So like over here, heat sensitive liquids and gases, your IV bags, saline water, you'd use more filtration style sterilizing. Whereas your gloves, needles, catheters, you want to use ionizing radiation. Okay, that's it for microbiology. Onto immunology. We'll start with innate immunity. So this is a very broad overview. If you have to take one thing away, it'll be innate versus adaptive. So innate immunity is very rapid, from immediate to within hours, whereas adaptive takes a few days. Innate is pretty much very broad spectrum, so it's not specific. It doesn't change based on specific pathogens, which is completely the opposite. In adaptive, it's extremely specific, so it recognizes specific traits around specific pathogens using a bunch of receptors, using a wide variety of receptors. And then innate always has the same response because it's broad spectrum, so it's not specified, so it'll react the same way, and it doesn't have any lasting protection. On the other hand, adaptive, their response will vary according to the type of pathogen, so we'll talk about these, and then as with repeated exposure, that response will improve. So that's the concept behind immunity and vaccinations, is response improves with repeated exposure. Those are targeting your adaptive. And then this is just a timeline, basically, of your entire immune system, very summarized. Okay, your barrier defenses, first line of your innate response. So you've got your physical barriers, which is your skin, biggest immunological barrier. Your mucous membranes, coughing, vomiting, low pH in the gut, flushing of the urinary tract. These are all preventing pathogens from entering the body. Your chemical barriers are in your tears, lysozyme, and fatty acids on the skin. These help prevent bacteria from thriving. And then your normal gut, normal microbiome, normal flora that we talked about earlier. Ooh, this is here early. Okay, we'll come back to this. Oh, never mind. Okay. Okay, so your complement is basically your second line of your innate response. And complement are serum proteins, and they have these three different pathways, which is the lectin pathway, alternative pathway, and antibody pathway, which is I think the classical pathway on this diagram. They've asked like one question in the past about C3A and 5A, and it causes inflammation. So it doesn't hurt to know these specific things. It's not the highest yield information, knowing these very specific complement proteins. But yeah, these are the functions of complement, so what we're talking about here. So opsonization is the proteins that the complement proteins bind to the pathogen, and they act like a marker, a beacon to phagocytes to come. Talk about phagocytes later. Chemotaxis is essentially a signaling function of complement. Is it attracts macrophages and neutrophils to cytokines and chemokines. Cell lysis is via membrane attack complexes, and essentially just punches holes in the membranes. And if the membrane of a bacteria isn't intact, it'll obviously cause it to die. And agglutination is complement and antibodies cluster the pathogens together. So if you have a big chunk of many pathogens, it'll make it easier to phagocytose them. All right. These are pretty much an overview of all the cells you want to know regarding immune function. The ones in green are your innate immune system, and we'll talk about them. And then you've got your lymphocytes, your T lymphocytes, B lymphocytes, and plasma cells here. They've asked a question about like common lymphoid progenitors before. So it makes sense that your lymphocytes are coming from your lymphoid progenitor, but just keep in mind that NK cells as well are also lymphocytes in a way, and so they come from this embryonic origin, and everything else just comes from myeloid, and they all come from these multi-potential hematopoietic stem cells. Okay, now going through innate cells. Your phagocytes, you've got your three main types here: neutrophils, monocytes, macrophages. Your neutrophils are your most abundant white blood cell in the body. They're usually the first responders there. They're involved in acute infection. Sorry. And pus is primarily made up of neutrophils. Oh, sorry. The pus that's released in infections primarily has neutrophils and then dead pathogens and tissue cells. Monocytes are antigen presenting cells, which we'll talk about later as well. And they phagocytose antibody coated pathogens. And macrophages are basically a monocyte, so monocytes are known as monocytes in the blood, and when they move from blood to the tissue, they're known as macrophages. The other innate cells that you want to know are NK cells, eosinophils, basophils, and mast cells. Natural killer cells release pretty much are very cytotoxic. So they release these granules to kill cells that don't have a normal MHC1. And they kill cells with perforin, which again makes holes in the cell membrane. Eosinophils are for helminths, so important for parasitic infections. Basophils mediate inflammation, so they release histamines, and as well as mast cells, which are helping with inflammatory response as well. So these are both these three granulocytes. Yep. PAMPs and PRRs. So pathogen-associated molecular patterns. So these are pretty much how our immune system detects whether or not something is a pathogen or is foreign to us. And so these are your like lipopolysaccharides, your flagella, double-stranded RNA. All these structures are not on our human cells, but are on pathogen cells. And so when we detect these patterns via our pattern recognition receptors, that's when we know to start an immune response. And a key important fact is that the PAMPs are actually important for pathogen survival. If they weren't necessary, through evolution, the pathogens would just evolve away from having them, because the pathogens that don't have these PAMPs would, if they would survive, then they would all just evolve to not have them. Whereas because they're important for pathogen survival, they're necessary in pathogens, and then we can use those to target pathogens. Pattern recognition receptors, your main ones that you want to know are toll-like receptors. So these are receptors in our body that detect PAMPs and then start the immune function. Okay, how these interact is on when toll-like receptors or pattern recognition receptors interact with the PAMP, that's what causes phagocytosis. So these cells that the phagocyte cells, macrophages, monocytes, dendritic cells, and neutrophils have these pattern recognition toll-like receptors on them, and when they detect a PAMP, they will phagocytose and then break down the pathogen into these three enzymes: free radicals and hydrogen peroxide. But yeah, okay. So this is just going in depth into what we talked about here. Cool. So phagocytosis is where phagocytes engulf and destroy pathogens that are, or foreign substances. So these are your main 6 steps that you want to know. It starts with pseudopodia, which is Latin for for fake legs. And it's pretty much this area here where a phagosome will extend its cytoplasm around the pathogen and engulf it via endocytosis. And then it forms a vacuole, which has the pathogen inside it. And the vacuole with the pathogen is known as a phagosome. And then a phagosome and a lysosome fuse. A lysosome has your toxic compounds and enzymes that break down pathogens. So when the phagosome and the lysosome fuse, it forms a phagolysosome. And then the debris from breaking down the pathogens is released from the phagocyte by exocytosis. So pseudopodia engulf, creates a phagosome, fuses with a lysosome, which forms a phagolysosome, breaks it down, and then exocytosis. Okay, migration of leukocytes. So leukocytes are your immune cells, and they're normally in your blood, but when an infection happens, your pathogen's likely in your infected tissue or your lymphatics. And so your leukocytes need to migrate from the blood into the infected tissue. And so this is an overview of how the process happens, but I'll go through it here. So it's driven by histamine. So mast cells and basophils in the tissue or in circulation find the pathogen and release histamine. And what histamine does is it increases the permeability of endothelium. So endothelium are the cells lining your blood vessels. And as you can see, usually they're tightly packed, but if a molecule needs to pass through it, it needs to be more permeable. So tissue macrophages that have detected the PAMPs and DAMPs that we talked about, once they phagocytose the pathogen, they release these cytokines and chemokines, which are your communication molecules. And that increases the expression of surface selectin. So this whole process till now is known as endothelial activation. So selectin is just a molecule on the endothelium which is upregulated by these cytokines and chemokines. And so leukocyte has the alternate selectin ligand, which binds to selectin. Now that there's more selectin on the endothelium, normally this leukocyte would come near the endothelium and kind of just roll along it and then go off back. But when there's more selectin, the selectin ligand on the leukocyte will bind to the selectin on the endothelium. And then when the selectin and selectin ligand bind, that increases the affinity of integrin. So integrin's on the cell, and integrin and selectin ligand are on the leukocytes, and then integrin ligand and selectin are on the tissue, on the endothelium cell. So when the affinity of integrins on the cell increases, it causes it to bind onto integrin ligands on the tissue. And then again, as we talked about, histamine increases the permeability. So now the leukocytes are here on the endothelium, and because it's more permeable, they can enter the tissue. And there'll be a chemokine gradient caused by the tissue macrophages. And so leukocytes just have to follow that chemokine gradient towards the pathogen. All right, so inflammation. These are pretty much your 5 main indicators of inflammation are your pain, swelling, loss of function, redness, and heat. And this is how they're caused. And that's it for innate immunity. Lastly, on to adaptive immunity. So this is more specific. So we'll talk about lymphatics first. So your lymph vessels are pretty much, they're these vessels that drain your tissues of excess fluid, and they pass through lymph nodes. And lymph nodes contain a lot of your B and T cells, your lymphocytes, which are involved in adaptive immunity. And so when your antigen presenting cells, which is dendritic cells, which we'll talk about on the next slide, they go through these lymph vessels into the lymph nodes in order to activate the adaptive immunity. So your primary lymph organs where immune cells are made are your bone marrow and thymus. And so B cells are made in the bone marrow because B and B, and T cells are made in the thymus. And then your secondary lymphatic organs are your spleen and your lymphatic just drainage. Yep, that's pretty much the same thing over there. So just a general overview of different lymphatic things, but just know and then there's also like tonsils and different organs, Peyer's patch, that are involved in secondary lymphatics, but just know the spleen, pretty much it's like lymphatics for the blood. Okay, dendritic cells are your first way of getting from the innate immune system to the adaptive immune system. So it is an antigen presenting cell. It phagocytoses pathogens. And once it has those pathogens, it'll then travel to the draining lymph node. And over there you have your T and B cells. And so there it'll activate a naive T cell. And this is a complicated process we'll just go through now. And that once the T cell is activated, you've now got your adaptive immune response, and you'll start making, your T and B cells will start making specific targeted responses to that pathogen. MHC, all right, major histocompatibility complex are proteins on the surfaces of antigen presenting cells, and you have these 2 classes. So all cells in the human body have your MHC class 1, and that's because it presents intracellular molecules. So anything on the inside of a cell will be expressed by MHC class 1. So think viruses, cancer, that's where it's important. And so these will interact with the CD8 plus and natural killer cells. CD8 plus is like interacts when there's a virus on the MHC class 1, and then natural killer cells is like when there isn't an MHC class 1 of a normal, because cells can present normal molecules inside them, just it's kind of like telling the environment what's going on inside. And so if there's something wrong, it might pull and there might not be anything. So CD8 is when there is something wrong on an MHC class 1, and natural killer cells is when there's an absence of MHC1. And MHC class 2 goes with your CD4 plus cells, and it's only expressed in your antigen presenting cells because it's presenting foreign molecules. So it has to first phagocytose pathogens and then break them apart and then present those proteins on MHC class 2. So think of the rule of 8 as just like if you multiply, it has to be 8. So 1 times 1 goes with 8 because 1 times 8 is 8, and 2 goes with 4 because 2 times 4 is 8. All right. So your lymphocytes, your major lymphocytes are your B lymphocytes and your T lymphocytes, and they originate from bone marrow. So they both originate in the bone marrow, but where they mature is dependent on which type of cell. So B cells mature in the bone, and T cells mature in the thymus. And yeah, pretty much once they're mature, they go around in the blood and the lymph until they're required. Okay, this is a very broad overview of basically your highest yield stuff you need to know about your adaptive immune system. So we've got the dendritic cells that we talked about, and they move to lymph nodes, and they activate this naive T cell. So naive, and then when these T cells are activated, they differentiate into, or normally they differentiate into CD8 T cells and CD4 T cells. And CD8 T cells can be activated by your MHC class 1 that we talked about, and then it becomes a cytotoxic cell and destroys infected cells. So this is your cell-mediated response. You have your cell-mediated and your humoral response. So with CD8, it's more of your cell-mediated response where the cells are actually, your CD8 T cells are going around and destroying cells. And your CD4 T cells are presented, are activated by antigens presented on an MHC class 2. And so they become T helper cells. And they don't do much by themselves, but they help enhance the immune response. So they activate B cells, they activate natural killer cells, and other phagocytes. And so when the T helper cells activate B cells to proliferate, those B cells then can form either memory B cells, which forms memory of a specific antigen, or so that in the future, infection can be quickly resolved, which is how the adaptive immune system gets better and better every time. Or it can cause plasma B cells, which is your main B cell in order to secrete antibodies for the current infection. And then what's this last thing? So your antibody receptor recognizes specific antigens. Yeah, so this is pretty much just about how your antibodies and your B cells interact. Co-stimulation. So we talked about how T cells can be activated. So all T cells require 2 signals to be activated. It's just a little, it's a little thing that Monash loves to test, but basically the MHC and peptide combo from the dendritic cell or the antigen presenting cell and another protein. So then you need 2 signals to be activated. Oh, I missed something there. And then when these cells are interacting, there's B cell receptors and T cell receptors. And these receptors are very specific to the particular antigen, which gives the adaptive immune response its specificity. And the variable region that we'll talk about later is what's responsible for the specificity. That is in a few slides. I'll talk about that more. Okay, so over here, we had the creation of a CD4 T cell. So CD4, the cytokines that are the cytokines on the CD4 T cell influence the type of T helper cell it becomes. So we had CD4 cells, but the CD4 cells can differentiate into even more different types of cells. So if there's IF and gamma and IL-12, it'll turn into a T helper 1 cell. And T helper 1 cells are primarily for viruses and intracellular pathogens, whereas if there's IL-4 and IL-5, which are just cytokines, it'll turn into a T helper 2 cell, which is more for helminths and allergens. And these are just the ways it acts. Okay, now when the B cells are activated and plasma B cells are made, they produce antibodies. You've got these 5 major types of antibodies. Honestly, the highest yield things to take out of this are your IgM is your first response and your IgG is your later response. So just remember M comes before G. And your IgG is can cross the placenta, so it's responsible for passive immunity in the fetus. In terms of pregnancies and when a baby is born, they have that IgG as its main source of immunity. IgA is mucosal, and the rest is not too relevant. IgE was involved with allergies. Yeah. Okay. So I was talking about earlier how your T cell receptors and B cell receptors, those are the primary ways that specificity is achieved in the adaptive immune system. And so you have these variable regions, and because your T cell receptors and B cell receptors are basically proteins, so they're determined by the DNA coding for these receptors. And so when DNA recombination occurs, it creates all these different variable regions. And so when one specific type of receptor is activated, they'll launch an immune response towards a very specific pathogen. So you got these V, D, and J segments that are pretty much the main way of causing differentiation and specificity. Okay, more on antibodies, how they function. So they have these three major ways of working in the immune response. So one is neutralization, which is preventing the interaction of the actual bacterial toxin with the host cell. So this is more for like viruses and toxins from bacteria that need host cells to cause damage. If you prevent that interaction, it won't occur, like damage won't occur. Opsonization is pretty much, when you think of opsonization, just think of like coating the pathogen. So antibodies can completely coat the pathogen, and then that becomes like a marker for phagocytes to come and phagocytose it. And the antibodies can also activate complement. So coat the pathogen and it'll form a receptor for a protein from the complement system. And so when the complement binds, the complement can either kill the pathogen, which is the membrane attack complex that we talked about, where it can like make these holes in the membrane, or it can further opsonize the pathogen, coat it, and then make it ready for when phagocytes come. Acquired immunity, you got these four main types. This has been tested quite a bit before. Just know your natural versus your artificial immunity. So natural, obviously with no like pharmaceuticals or anything, your most active natural way of getting immunity is just by infection. So when your adaptive immune system is activated, it'll have those memory B cells. And so you have that immunity if that pathogen was to come again. Natural but passive immunity is when you haven't actually created the antibodies. So these are pretty much in babies and fetuses where they get these immunoglobulins or these antibodies from the mother. So specifically IgG from the placenta or IgA from the breast milk. In terms of artificial, active artificial is when you get a vaccine. So even though you're getting a vaccination, you still have to initiate the immune response, but you haven't gotten the pathogen via like just normal day-to-day stuff. You get the pathogen through vaccination. And then artificial and passive is when you don't actually have to even activate your immune system. You just get antibodies injected into your serum, such as your antitoxins. I think this is the last slide. Yep. Okay. So monoclonal antibodies are a drug treatment. And this is regarding your last artificial and passive immunity. So pretty much the way it's used a lot in cancer treatment to prevent immunosuppression. And pretty much what it does is like you use an animal like a mouse and you present the pathogen's antigen into the mouse, and the animal's immune response will be activated. It'll create these plasma B cells, which you can then harvest. And you also take tumor cells. And because the main function of tumor cells is they continuously replicate and they can kind of immortalize the plasma cells. So you join these and you create this hybridoma, which is basically the best of both worlds, where you have these plasma cells that are making antibodies and can replicate forever. And you have, and then you kind of just collect the antibodies, and those are your monoclonal antibodies, which when you inject into a patient, they don't actually have to make an immune response because the antibodies are already created and they can fight the infection. All right, how are we on time? Ooh, it's 1:30. I don't think you guys got a break. Bhuvi, how are we on time? Because there's like a heap of questions. How do I stop sharing? Okay. There's all these questions. I think it was probably best if you go through them and you're on time, but they're really useful. I think questions are probably one of the best ways to study. So you have the slides. I'd recommend going through these questions and you're on time. And let me just check the chat if there's any questions. Okay. Where do dendritic cells originate from? That's a good question. Is it in the diagram? It's not in the diagram. I'm not sure as of now. I can get back to you. I will take a screenshot of this so I don't forget. Okay. Where does CD8 T cell activation occur? Does it occur in lymph nodes? And if so, how do infected cells get there? So in fact, the way CD8 T cell activate, so it occurs in lymph nodes, first of all, and it occurs by the way it gets there is dendritic cells that are activated all across the body, which are your phagocytes. They migrate through the lymph and they travel to the lymph nodes. And when the dendritic cells interact with the T cells, that's when CD8 T cells are created. Which scenario results in negative selection of a CD8 plus T cell during development? Okay. I'm going to share my screen again. So this is talking about one of the questions that are there. Not sure if I have that diagram. It pretty much, okay, I'll go to the question. Let me find it. Okay. So which scenario results in a negative selection of a CD8 plus T cell during development? So if we think about what CD8 plus T cells do is they interact with the class 1 MHC markers because of the rule of 8. And so a strong affinity for a class 1 MHC marker it's pretty much how CD8 plus T cells are created when they're differentiating from normal T cells. A T cell that has a stronger affinity for a class 1 MHC marker is more likely to differentiate into a CD8 T cell. There is a diagram for this that explains it a lot better than I could explain it in words. And I'll just add it onto this answer sheet right after on this answer slide. So when you're going through the questions, you have a better explanation. But it's pretty much, you think of the role of CD8 T cells and then you see which option fits with that better. How much depth do we need to know about lymph node structure? I'm not sure what you mean by lymph node structure. I don't really know what lymph node structure is, so I wouldn't guess too much. Just know that they're like pretty much just areas in lymphatic vessels in the body where a lot of T cells and B cells are. Do the T cells activate B cells or the other way around or both? T cells activate B cells, yeah. So no, you're correct, yeah. T cells activate B cells. It's not the other way around. B cells create antibodies, which then go on to cause the immune response. But it's like dendritic cells to T cells to B cells. Which of the following signaling molecules, cytokines, are pro-inflammatory and increase vascular permeability in the site of infection? I'll go through this as well. I'm going to take a screenshot and then I'll get back to you. I'm not too sure, sorry. B cells can receive stuff from the receptors. I believe I'm pretty sure they can actually receive signals from antigen presenting cells. I'm not sure if it's been tested, but it's not too high yield. The main way you want to know is like, if you understand this diagram, you'll pretty much know all of what you need to know. But I think there is a mechanism through which B cells can get activated by antigen presenting cells. I just don't know enough about it. Answer on slide 115. Okay. Because I remember having issues with this question as well last year, and I kind of just copied and pasted all the questions. Yeah, okay. Honestly, just don't discount this question because I always thought varicella zoster also caused shingles, and I'm not sure why it was used as that. I kind of never got my answer last year. But pretty much HIV does cause AIDS, hepatitis B causes liver cirrhosis, Epstein-Barr virus causes glandular fever, and HPV does cause cervical cancer. I'm pretty sure varicella zoster causes shingles. I'm not sure why it's wrong there. Is there any question I missed? There's quite a few, sorry. I think that's all. I'll go through this questions I screenshotted and try to get back to you. Yep. Okay. I think that's all for today.
Thank you, Yajat. And thanks to everyone who came, watched the revision lectures. So as Yajat said, the slides are up on the Mumis Drive, so you can have a look at his questions and like all the other slides up there. As well as the recordings, which will go up hopefully later today. The year one recording's up. Oh, sorry, the day one recording's up already. So if you guys wanted to look back at that, it's all there. But yeah, a huge thanks to Yajat and all of our other presenters and revision lecturers. They really made these revision lectures amazing. So thanks to everyone for coming as well.